kind introduction. Can everybody hear me well? Sounds good. Okay. Yeah, I'm really excited to be here. It's been a wonderful meeting already on the first day. I'm very sad also that I will have to leave this evening because I'm organizing another meeting in Berlin, the Biomembrane Days. You should all come one time. I'm really sad I'm going to miss tomorrow's and Tuesday's talks. And I want to thank Ali and Silvio for making this happen. This is much appreciated, much needed, and I hope you can bore a tradition maybe on the last evening with a couple of beers of coercing somebody of doing it next time, where I'll be glad to attend again. So bear with me today because I have three unpublished stories that I want to share, so I'm going to be a bit fast. I hope that's okay with you. So I'm a cell biologist myself, and we are interested in cell biological problems, obviously, and towards that aim, we develop microscopy approaches a lot. So for example, we make our own 3D printed microscopes to put into incubators. We have recently developed a method to optogenetically release single molecules in a dose-dependent manner, which we believe will be especially important in the context of min-flux microscopy, where you need to have very few molecules present at a time. We also used LDNA for DNA paint, and this allows you to do DNA paint in the presence of a lot of endogenous DNA, like in the nucleus, where left-handed DNA cannot bind to the endogenous DNA. Now, the biological problems we're interested in are viral membrane dynamics, so we like to understand how viruses interact with their receptors and how they can then internalize cells, especially in Clathrin-independent manners. One further project concerns membrane compartmentalization, so we try to understand how molecules at the plasma membrane are confined and controlled in their motion by the submembrane actin cytoskeleton, and here we use the paradigm of the actin rings in neuronal cells to understand how that can be brought about in live cells. The main interests are septins. These are an enigmatic and poorly understood protein family that are especially interesting for single molecule biologists because they are complex. The subunits of their filaments is exactly 30 nanometers long with the same molecule on both ends. So if you label it, we can here really see the assembly of the whole filament from subunits, both in filamentous fungi as well as in mammalian genome-edited cells. And I'm really excited that tomorrow you're going to hear more about septins from Sushavan Chanda, who used the fabulous one microscopy from Ali and Silvio, and he's going to present some of our septin data hopefully on Monday. I'm very sad I'm going to miss that. Our interest in super resolution and single molecule microscopy stems from the frustration I experienced studying biochemistry in the textbook, where multi-protein complexes look like this. Yeah? So you have a lot of information contained here, of course. This is assembled from decades worth of biochemical and structural biology work, where individual components and interactions have been identified and sometimes detailed down to the structural level in structural biology. However, it's clear that this is not how multi-protein complexes look, right? In the focal adhesion, you don't have really a tail end going to the left with 3 vinculins on top, right? That's not how things work, but rather it looks like we have modules assembled in a way that they can execute the same function in different complexes to make a plethora of functions from a limited genome. And a wonderful example here comes from Ksena work, where he looked at the very focal adhesion and could identify layers or modules in which individual proteins and functions would be concentrated, and some of these proteins would be organized to execute different functions at different positions. And I think this is how we in the future will understand more about the functions executed by multi-protein complexes, especially together with multiplexing, as we heard about yesterday from Felipe and others, and the high resolution as Markus Sauer and Jörg Beversdorf introduced us to yesterday. So what we want to do is we want to see all the molecules at the native relative position to one another, right? And this was the aim we had at the onset of my lab in 2009. And we built a single molecule microscope with the aim to understand where many molecules are relative to one another. And very quickly it became apparent to us that if we use antibodies, this is not really the way to go because you're going to label all over the place, not where you want to. And at the time, there was a new tool, the GFP nanobody by Uli Rothbauer. And by putting a dye on there, we quickly realized we could circumvent the so-called labeling error. And of course, in Göttingen, I don't have to introduce that this is a very, very useful concept and The Gerlich, Opaso, and Risoli lab, of course, have expanded this to an absolutely fantastic state where you have multiple startups, and now we can label, as you've seen from Philippe's work, 8, 10, 12, however many proteins in multiplex imaging. Now, to drive that further with expansion microscopy, we try to bring the dye even more closely, and to do so, we combined a benzyl guanine with a fluorescent dye and an acrylate moiety, right? Thinking that, well, now we can link our dye directly into the gel, directly on our protein. So our organic chemists here at FU in the Haag lab, they made that for us. We were really excited. We expressed the SNAP-tag protein, went into the cell, stained it, made the gel, digested it, and then we saw this. Okay, not exactly this. I don't want to show you fake data. Yeah, this is just a black slide. But this is what we saw repeatedly, and we grew frustrated and more frustrated. We didn't really know what to do. So we went all the way back to the cuvette and mixed our component with increasingly complex mixtures, with APS, with TMAD, just with the gelation mixture. Eventually polymerized the gel inside the cuvette to see if something's wrong with our dye. Nothing was wrong with our dye. And we didn't really know what to do. So then we did, okay, we're going to do one last experiment. We're just going to add ACX, okay? It's counter to all we wanted to achieve because ACX should have been already on our dye, but it helped, yeah? So here, after gelation, the dye is fine, whether we add very little ACX or a lot. But then, after expansion, only if we add fairly high amounts of ACX, we can see our dye. So what does that mean? How does that make sense? Again, we talked to our chemist friends after we quantified this, so really dose-dependent labeling after ACX addition and not if you don't have the acrylate on the dye. Yeah? So both are required, ACX and the acrylate on the dye. And the chemist then explained to us, well, this is a very clear phenomenon to every chemist because this depends on grafting. Yeah? That means that if you initiate polymerization, you have these growing ends in solution. But because there is a very high concentration of diffusing monomers, the likelihood to connect to a freely diffusing monomer is so much higher than to one of the sparse ones that are on your target that very likely they're not going to be incorporated. So what you have to do is you have to add more monomers onto your target structure, and then this will lead to something called the grafting through, which is basically you make contact, and then the degrees of freedom of your growing chain is reduced, and you're going to finally incorporate also your other surface-connected monomers. Okay. So of course we wanted to test that, so we expressed a Clathrin light chain with a SNAP tag and a GFP. Okay, so now on the SNAP tag, we put our dye with the acrylate moiety on it. And on the GFP, we add an anti-GFP nanobody that had an ACX on it, right? So we add extra monomers just to the structure we are interested in. And if we did that, we could rescue staining. We have wonderful staining. And if we added the ACX to stain microtubules, we could not do that, okay? So you need to deliver additional moieties just to your target structure to enhance grafting of your dye into the target, okay? So the chemists, whether you are bored, for us this was really an experience, and I think it helps us to think about how expansion works at the molecular detail. A couple of years ago, we also tried to use expansion microscopy with STED and adding a lot of dyes. This worked really, really well. You can see here the specifications of how we did it. The only drawback here being that you really need a long time. Yeah? So this whole cell here took 8 hours to image at 25 nanometer voxels. So I cannot really recommend that on a daily basis, and that's why people like to look at tinier structures. But the resolution is really fantastic. Here you can see the XZ cut. You can separate microtubules that are really only a few dozens of nanometers apart. And then we could also look at cilia. You can see here the microtubule doublets, but of course this is nothing compared to what Paul and Virginie are going to show to us in the next days at the example of the centriole. So now I want to go to some unpublished data and go back to septins, which have been discovered in the famous Slater Nobel Prize winning screen by Lee Hartwell, in which all the CDC, cell division cycle mutants, have been discovered. Okay? So CDC 3, 10, 11, and 12 had the same phenotype, which was they could not upsize the divided cell. Okay? Later it was shown that these molecules, then called septins, were localized to the septum, to the mother bud neck, in these electron-dense structures. And they then separate into two rings and the two daughter cells, and this is conserved into mammalian cells. So this is a rat cell. And you can see that it localizes here to the constricting, where we can't really see that, unfortunately. Here now you can see it, but it also has functions later in the cell division cycle. Yeah? And what it does do there, it binds to pips at the membrane, it binds to anilin, it binds to myosin, and later on it recruits ESCRT. Okay, so there's many septin-dependent functions in the whole process of cell division. Septins are indeed absolutely essential for cell division, and this was finally proven just this year when people did a maternal knockout. So in the oocyte, there's no septins left. Yeah. And you can appreciate here that after 3 days, a normal oocyte would already, embryo would already undergo gastrulation, whereas here you're still struggling with the very first cell division. Okay, so the very first cell division of an embryo fails if you don't have septins, and this can be rescued by injection of septin 11 RNA. Why this is particularly interesting to us is also that there are cells in the human body that do not need septins for cytokinesis, and these are cells of the hematopoietic system. So now this is of course very interesting because targeting septins by small molecule drugs may now lead to chemotherapy that do not lead to immunosuppression and destruction of the blood stem cells, okay, saving expensive stem cell therapy and immunosuppression. And that is one avenue we aim to pursue as well as a cancer therapy that does not kill the immune system. But back to the project, this was done by Nadia, absolutely amazing student in productivity and skill. And you can see here the doodles she made on the contribution of septins to cytokinesis. So they help in assembly of the contractile machinery and cleavage furrow constriction, in intercellular bridge formation, mid-body formation, intercellular bridge maturation, and finally abscission. Now, why do we need expansion microscopy to look at cytokinesis? Well, it has, in my view, never really been looked very, very well at cytokinesis, and this has reasons in the process itself. Yeah, this is a process that takes a long time, about an hour and a little longer, but within that time you have very fast three-dimensional processes such as the constriction of the cleavage furrow that you can barely observe here, and then a process that takes very long, which is maturation of the mid-body bridge until abscission. And within that time you have several processes happening and molecular multiprotein complexes assembling and disassembling and doing their work that are not really organized very clearly, and there's no way of doing volumetric, even less super resolution imaging over this entire process over high resolution, right? And that's why we decided we want to do expansion microscopy of thousands of cells over that process and then generate pseudo-time progression. So first we needed to identify features that we can use to determine where we are. And we started by using lattice light sheet microscopy because here you can really look at plenty of cell division events in real time. We could then extract the relative state these cells are in and easily image dozens of cells over that process. We could see that our cells are very happy and healthy. It's very reproducible how long the first stages of constriction and spreading and septin ring split take. Only then until abscission it takes a little bit of a longer time. And what we could easily observe just by extracting microtubule thickness, you can see here. So first it's a thick microtubule bundle as the spindle is compressed, and then the mid-body here emerges, and you can see there's a steady decline in thickness also over these stages, right? But what you can also see here, by all the individual lines, every line here is one cell terminated at the last image after abscission, is that just from microtubule thickness alone, you cannot deduce to the relative time where you are in cytokinetic progression. So we decided we're going to use the very feature-rich staining of septins all over progression of cytokinesis in combination with a higher resolution of expansion microscopy to extract features that allow us to determine where exactly we are in a still image during the process of cytokinesis. Okay. So we turned to expansion microscopy, and here you can see again why this was eventually conceived by Ed Boyden, as I understand it, is that here there's a black line if you do antibody staining of the cytokinetic bridge. Yeah? And that's not because there's no microtubules, quite the contrary. That is this area down here, and that is so black because not only are there overlapping microtubule bundles from the two cells, but also there's a huge number of other proteins that make this mid-body, and this is just too dense for antibodies to access, right? And this is very clear if you then do expansion microscopy, and here you can even see the bright staining here right in the middle, and this is where we have more tubulin, because this is where the microtubules overlap. Okay. So by expansion microscopy, we get access now to staining the mid-body. So Nadia then took thousands of images over from synchronized cells that we released for different time points, and then she could easily classify into 6 distinct stages: round and constricted right after constriction, cell spreading when the cells start to assume their shape again, then the septin ring split where septins move apart, septin manchettes where basically the intercellular bridge emerged and it's just sticking sort of out of the cells, and some septins left in the middle, the before abscission stage where we have a thin elongated mid-body, and then finally, obviously, abscission, here defined as microtubule severing, but there can be some membrane connections left, but we defined it as membrane severing here. Now, combining microtubule and cell shape and septin features, we could then do a principal component analysis that allows us to visualize cell cycle progression over time. And you can see here that round and constriction cell spreading, we could not separate so well, and that's because in the expanded cells, oftentimes we don't see the entire cell, which makes it different to recognize, but the ring split, septin manchettes, and before abscission stages are clearly defined, and that's very exciting for us because especially the septin manchette stage has not really been recognized as a specific cell cytokinetic stage beforehand, and it's neatly snuggled here in between ring split and before abscission. Now, following the pseudo-time progression line, we can then create a movie of expansion super resolved cytokinetic progression over time. This is 1,500 images averaged into 34 frames by moving average, and you can appreciate here how a tube of septins then moves to the outside and leaves a little bit of septin in the ring behind. Now, we did not only stain septin and tubulin, but always carried another marker. And this may be another septin or Borg, a mid-body protein, actomyosin aniline components, or parts of the ESCRT complex that are involved in cytokinetic abscission. And we could thus now recreate temporal progression at super resolution for all of these structures. You can see that here exemplified in MacRec gap, and you see staining of the very mid-body, and here you can see assembly of ESCRT from very early assembly to finally making these cones right before abscission. And I'm going to go through just a few examples to show what we can see now that was not accessible before. And here you can see again the stages from round and constricted, cell spreading, septin ring split, septin manchettes to before abscission. And one thing that immediately became obvious is that very early on, myosin B is already split into 2 rings while septin are together. And secondly, myosin 2A is always found cellward from the myosin 2B, okay, which is still more to the inside. Here you can see myosin 2A is cellward distributed, and very quickly it assumes already reorganization of the cell cortex, while myosin moves on to stick to the myosin, to the tubulin bundle, whereas actin supports integrity of the intercellular bridge. If we now make a cut through the mid-body, we can see that septins remain always on the membrane, whereas myosin undergoes a clear shift from membrane-associated to tubulin-associated in the cell-rounded cell spreading stage already, and particularly strong in the septin manchettes stage, ring split and septin manchette stage of cell division. If we look at septin and aniline, we can see a similar progression from a tubular staining where aniline is at the membrane together with septin, right in the middle of the intercellular bridge, whereas later it then moves along the tubulin and even the mid-body bulge as stained here by MacRec gap 1, whereas septin remains on the outside of the mid-body. Again, we can cut through right in the middle and a little bit laterally on the bridge, and you can see it starts a little bit here, but then in the septin manchette stage, aniline is both in the very mid-body and laterally on the microtubule bundle associated with tubulin and the midbody, while septin remains behind on the outside. With the central midbody proteins, we looked at citron kinase, which is a row kinase involved in spindle formation, PRC1, which is a microtubule cross-linking protein that cross-links opposing microtubules in the spindle and later in the midbody, and then macragap1 and MKLP1, which together are part of the central spindle complex. And you can see here that they all localize to the central spindle, whereas citron kinase forms part of the bulbous midbody protein, and PRC1 stays with the ever-thinning microtubule bundle. You can see this wonderfully as we cut through this, where PRC1 is always inside of the septin staining and then becomes spread out on the tubulin, whereas MKLP1 forms this bulbous head that emanates above that together with MKLP1 and citron kinase. Here you can see that as well. PRC1 becomes ever tinier with the tiny tubulin bundle, whereas citron kinase and especially macrogap and MKLP1 form this bulbous big midbody bulge. What we found particularly interesting here is that PRC1 consistently forms this little dotty pattern, which makes a lot of sense because these are individual cross-linked microtubules that are stuck together by PRC1 complexes. What is a bit more strange is that citron kinase, which is a membrane-opposed row GTPase, is here found in a dotty pattern, which we would not expect. But maybe this could be vesicles that are carrying row citron kinase here. Of course, the final abscission is done by ESCRT. You can see here TSG101. This is an ESCRT1 molecule. Consistent with that, it's assembled already early on and then later recruits IST1, which is an ESCRT3 subunit. We can see it already in the septin manchet stage in either 1, 2, or 3 rings. With 3 rings, we name everything that has already one cone on one side. But it becomes more prominent in the before abscission stage, where sometimes we can even see 2 fully formed cones on both sides of the midbody. Okay, this is the number of individual stainees you can see here. This is the quantification. So the septin manchet stage we can sometimes see, but in most cases we do not see either CHIM4B or IST1 subunits of the ESCRT3 complex, whereas in the before abscission stage we can see reliably ESCRT being assembled here. Okay. Strikingly, when we have 3 ESCRTs, we see this little dip here in the tubulin bundle showing the site of future microtubule severage. And when we then look through time after synchronization release, we can see consistent with our determining this as a distinct cytokinetic stage. At the beginning, we only see the septin manchet stage with 1 or 2, you can see that as these lines here, ESCRT structures, and then later only the before abscission stage appears with more ESCRT structures available. After 60 minutes, we already have abscissed cells and consistently more ESCRT around, and then that trend continues. If we look at the quantification this way, we can really see that with the bins, or actually starting from the top here, with the bins of time in our movie, we can first see the septin manchet stage, then the before abscission, and we have a temporal progression with the assembly of ESCRT, further supporting that the pseudo-time we generate from the principal component analysis rightfully recapitulates pseudokinetic progression. Now, Nadia made unbelievable visualizations of all the data we gathered. Here you can see the UXM individual staining representing the individual stages. These are the doodles of where the relative proteins are from the early constriction to rounded constricted, cell spreading, septin ring split, septin manchets before abscission and abscission. This is the averaged images in the top view of the respective structure, here membrane and in cell mask and tubulin, and this is the exoradial view. Now I'm not going to go through all the markers, septin, actin, and lilin, and so on that we have presented here, but I just want to go through with you a little bit through what we learned from this. This is now out on bioRxiv. We have there also a file in which we have all the markers over the 6 stages in X, Z, Y, UXM. So you can look at your favorite cytokinetic protein and do your own overlaps to study what protein interacts with which one. So the most striking things we saw was the membrane to mid-body transition of citron kinase early on as cells spread, and then later the mid-body bulging already in the septin manchet stage of citron kinase. We observed a contractile ring split of myosin 2B early on, and that was actually much stronger for myosin 2A that already spread in the septin ring split stage to the back to the cellular cortex. We saw that anilin undergoes a membrane to mid-body transition as soon as the septin rings split and very pronounced in the septin manchet stage. And the same holds true for actin, that though remains at the membrane cortex. PRC1 would spread along the microtubule ends in the septin manchet stage and later, and centrospindlin would contribute to mid-body bulging only from the septin manchet stage on. And we saw from the septin manchet stage on assembly of IST1. Now, Nadia assembled all of these markers in beautiful pictures. It's really not easy, as Felipe can probably sing you a song about that, to assemble plenty of markers into a single image. Please feel free to appreciate that. It's now out on bioRxiv as of yesterday. So we hope this is going to be published really soon. And we want to continue investigating this, going even higher in resolution. So I brought 2 more stories I would like to share with you. One is really for the lovers of abbreviations. This is UNROOF PAN U STED XM. Okay, this is Pan Ultrastructural Expansion STED Microscopy of Membrane Sheets. Okay. So what we did here is we generated membrane sheets. So we cultured cells, and with buffer we blew away the top of the cells and the nuclei. And thereby, all we have left over is basically the bottom leaflet of the cell. Okay, we can then just gelate and expand and do a pan staining. And this is very similar to what you know from John Heuser's work and Justin Tarasca's work and Stefan Vassilopoulos and Christophe Leterrier's work on neurons and many others, where you can really look at the top of the inside of the plasma membrane. You can really nicely see these Clathrin-coated pits at different stages of invagination. You can see actin fibers. You can see little caveolae striated here in this platinum replica yam. Okay, and now we wanted to see if similar work is possible using expansion microscopy. And already when we did the first stainings, you could see here in confocal and STED microscopy, in expansion already in confocal, we had these very prominent dots popping up. And then in 2D STED, we saw that these are actually really rings. So we thought, whoa, this looks really like anocytic entities. So let's look a little closer. And we did 2D and 3D STED. Here you can see such a cell. I'm going to focus on these 3 tiny structures up here. This is what they look like in confocal. In 2D STED, you can really see nicely the individual shapes. This one's already very round and like a ripe Clathrin pit. Then in 3D STED, excitingly, this guy is just completely gone. Yeah? And that is because, of course, with the tiny 3D STED point spread function, we are already at the plasma membrane here, and this guy is already inside the cell a little bit. Okay, so what we see here is a flat lattice. We see a curved beginning Clathrin-coated pit, and we see one that is already ready to take off. And I bet you there's dynamin right here. Okay. So we then, of course, did what you have to do. We did co-staining with Clathrin, and indeed, as you can appreciate here, all of these guys here, they are positive for Clathrin antibody staining. Okay, so we can just by looking at the pan staining, we can easily identify Clathrin-coated pits. Indeed, when we look at 3D STED, we can see the flat, the curved, the domed, and the precision stage. And already, if we now correlate this to the 2D STED image of the very same structure, we were confident that we can extract the information on what stage we're in already from the 2D data, and we trained a model, and indeed, it's possible for us to segment and classify straightforward the flat, curved, domed, and precision vesicles from 2D STED images of these membrane sheets. So to further look into this, we made use of a visualization technique made by Justin Tarasco, who has beautifully used correlative platinum replica electron microscopy and STORM to look at the assembly of the machinery of Clathrin-coated pit scission. So if you FCHO2, for example, is sort of at the edge of a Clathrin coat. Epson 1 and 2 are found throughout the Clathrin pit, more on the inside. And then, of course, dynamin in the end pinches the vesicle off by forcing the two membranes together and forcing membrane fusion biophysically. How he did that was he basically averages and then draws a line over this pit with the perimeter being the 0 point, and then you have either your inside of the perimeter or outside of the perimeter. And when we now co-stain Clathrin and NHS ester, we have already a pretty good overlap, but there's space for accessory molecules here and here that may contribute to that staining. Okay. Now, if we stain for FCHO, we can detect this at the rim of the Clathrin-coated pit as expected. And if we stain for Epson 1 or Epson 2, we can see this throughout and a little bit on the inside of the Clathrin coat, as you can see here for both of these markers. When we then look for dynamin, this really recapitulates Clathrin-coated pit scission. So first we find it sort of on the outside, just gathering around Clathrin. Then we find it to assemble, and then eventually being really in a tiny ring at the center of the very Clathrin-coated pit. It's possible to use pan-ultrastructural expansion microscopy together with STED of membrane sheets to generate high throughput assays of investigation of Clathrin-coated pits. The only thing we need now, because this is as of now still a bit of work doing the STED microscopy, I'd be very happy to talk to Stefan or Ilaria on how we can automate the acquisition of STED in our membrane sheets to get a higher throughput of this. So that was the second story. We're just writing up this manuscript spearheaded by Merit, a master's student in the lab. And there's one more thing I would like to share with you that is as of yet unpublished. And this is a comparative cell atlas for pan-expansion microscopy and cryo-X-ray tomography. So we are privileged in Berlin that we have a synchrotron nearby that has an excellent soft X-ray microscope. The thing here is that you can use cryogenically frozen cells that are not manipulated in any way, and you can use the water window of 2 to 4 nanometer wavelength and use these soft X-rays to use the high intrinsic contrast cells have here. And you can also do tomographic imaging, which means you basically tilt your cell through the X-ray. I don't know if that's a good experience. And then you can see these volumetric images from which you can reconstruct stacks. Okay, so this is always transmitted through the entire cell stack here and then imaged. And here you get about 30 to 40 nanometer resolution with the maximal sample thickness that's well within a cell monolayer. And this is sort of a gold standard technique for ultrastructure visualization without any chemical fixation or staining or sectioning. And we decided, well, let's use pan-expansion microscopy that stains everything in the expanded cell to see what structures would be particularly well suited to look at and which ones not so much. And of course, most interesting is what's not so much, in a way. So just to compare the imaging modalities, so this is a pan XM staining. Please excuse me, Jörg, for the amateur execution of this. This is, of course, wonderful because in 1 hour we can get 20 stacks with a relatively high field of view in our spinning disks. It's pretty straightforward to do a lot of get a lot of data. In cryo, we need an hour to get about 1 or 2 tomograms with a much smaller field of view, pretty much the size you can see here in that image. But on the first glance, it seems that both methods are pretty well suited to visualize cellular structures. We were particularly interested in membrane stains because these are, in our experience, very difficult to achieve in expansion microscopy. And these are the strength of X-ray because membranes have very high contrast, as you can see here with the lipid droplets in X-ray microscopy. But we have to say that NHS esters together with fixable lipid dyes are actually quite suitable for robust overall ultrastructure visualization by UXM pan staining. So to give you an example where it doesn't work so well, I brought to you here something we've been studying in the corona pandemic, and this is lysosome-associated multilamellar lipid deposits that are a hallmark of drug-induced phospholipidosis, as you get them either in COVID infection or in treatment with antidepressants like fluoxetine. Okay, and these multilamellar bodies you can see here are very easily seen in X-ray microscopy as these dark-rimmed vesicles, and in transmission electron microscopy we can even see these individual lamellae of these multilamellar vesicles. In UXM, in our hands at least, they have been much more difficult to visualize because we often have these sort of empty halo kind of regions with some dark matter inside, and this maybe reflects a little bit what we heard about myelination, that it's still difficult to achieve efficient myelin staining in expansion microscopy in many cases. When we then compare, and we are preparing this for a publication, you can look in detail much more in there, but here's a little bit of an overview. If we look at plasma membrane, we can get really nice overall morphology in pan staining. In X-ray, we can still see a bit more details. The nucleus is unfortunately smaller than the field of view in X-ray, but we can see very fine structure in pan-XM as well. We can visualize ER tubules and sheets in X-ray microscopy. This is a little difficult, at least at the resolution we have in pan-XM, just because the cell is so busy, and protein doesn't give as good contrast in membranes in pan-XM, so it's a little hard to say. Mitochondria, cristae are easier to visualize in our hands using X-ray microscopy. Certainly in higher resolution this should also be possible, but the network is more easy to access in our hands in pan-expansion microscopy. The one thing where X-ray clearly is better is the classification of vesicles, that here we can already by eye distinguish which is a lysosome, an early endosome, a multivesicular body, and that is relatively hard in expansion microscopy. What we like to use pan-expansion microscopy for a lot, besides looking for biological data, is checking our reagents and our assays, because it's absolutely merciless when it comes to exposing errors in your protocol. Because if you have nuclear detachment, if you have cluster formation of proteins in membrane, if you have distortion of mitochondrial structure, if you have large or small tears in the membrane, you always see it in pan-XM. So if you want to survey your defects or test new sodium acrylate batches or your gel handling, I highly recommend to always run a pan-XM experiment at the site. Okay, so I've been rushing quite terribly, so I'm already done. So I want to thank who's been doing the work. Mangfei has already long left the lab. She's now a postdoc at the University of Beijing. Ria did the expansion STED. Ria did the staining of the SnapTag dye. Nadia has been doing unbelievably all the expansion cytokinesis work. She's just looking for postdocs, so she's absolutely outstanding candidate. So if you have a really, really good project that is bound to succeed, please contact her. I'm going to be really mad if she doesn't end up extremely successful. Okay. Micha is now a postdoc at Helmut Center Berlin, and they have an excellent cell biology team there. So if you want to try X-ray microscopy, which really has some unique capabilities, please feel free to contact Michael and Jim McNally, who's still there with a long experience in X-ray microscopy in cells. And Merit is a master's student that has spearheaded the development of the expansion microscopy of membrane sheets. These green guys, these are undergrads that have been contributed greatly. I want to also have a shout out to Hauke Werner, with whom we've done some expansion of myelination, which is under review, as is the expansion of cytokinesis together with Michael Kraus. And if you're looking for a postdoc, feel free to contact us. 4 of the 5 postdocs we had already have faculty positions, so, you know, maybe we can make it happen. And I want to thank we're very thankful for the very pitiful amount of funding we get, but we hope it's going to be more very soon. With that, I want to thank you, and I'm open to questions.
Thank you so much for keeping the time, which gives us ample time for questions. So I think there's one in the back. If you want to ask, please come to the microphone.
Can you also use expansion as a method to improve your X-ray microscopy resolution?
I don't think. So the contrast, of course, if you pull stuff apart, is not going to be improved. And I fear that the gel itself, you know, it's also a carbon-based, so you wouldn't be able to tell what's protein, what's gel. I don't think that's going to fly.
So you can use this one while you...
Good. Have you tried to compare with cryo-EXM? Because here the X-rays compare with chemically fixed, where you have a cryo-X-ray. And did you try a membrane marker? Because you can't see the same in NHS.
Yeah,
and
so cryo-UXM, of course, is wonderful. We tried it as well. We find it superior in membrane preservation to anything else we've tried. Yeah, so this is definitely the way to go.
Because you see those holes around these bodies, right? You have this empty space.
We haven't looked at that particular thing. We just tried it because we had the plunge freezer there.
Yeah.
And you know, it just came out, and we were like, "Oh, it looks really good." So, so we just tried it, and we found it works really well. I can really recommend it. It's really cool. But
it could be
it could be an alternative even for
an issue of fixation, right? I think it's difficult to know if what we see is due to the fixation or the expansion itself.
Yeah, I mean, I think it's really a little bit out there what we do with membrane structures. Yeah, I'm already thinking on like maybe during expansion adding more lipids that you like, you know, make more membrane as it grows, because of course membranes with their flexible and highly complex shape are really, really hard to preserve in expansion in any way, because at some point you have to expand and they just don't.
Hi, very nice talk and very beautiful pseudo-time movies for the cytokinesis. I was wondering if you know whether the specific sequence of the heterooligomeric units, like the different septins during that process, are different. Do you have an idea of how they are like during the different phases, or what kind of specific sequences they?
So that is something we are very much interested in. Actually, so there's 13 human septins. That makes it one of the things that make it hard working with septins. One thing is that they interact with membranes, actin and tubulin. So whenever you pull down something, they're going to be there. The other thing is that there's 13 of them, and they make heterooligomeric complexes. So it's very hard to say who does what. So we actually have devised now exchange assays where we always knock out one and exchange it for the other one, and we can even exchange subdomains of the septins for one another. And this is what we are pursuing right now to understand what septins are required for what, because there is evidence from knockout animals that some septin functions can be replaced by other septins. For example, post-mitotic cells don't express septin 2 anymore, but septin 5. And we found that, for example, in dendritic arborization, septin 5 can be replaced by septin 2 and 4, but not by septin 1. So this is definitely a really, really important, but really, really difficult problem. So I, as a tenured person, feel I have to kind of tackle that, even though it's a pain. Yeah.
Thank you.
Fantastic talk. Thank you very much.
Thank you, Virginie.
Beautiful application. I was wondering, in your atlas, so the last part, do you plan to use the different NHS ester that exist? Because, you know, they might mark, you said, for example, ER, you don't really see, but because there are others also, maybe sometimes from Joshua, you know, you have different, pine. Maybe it would be super nice to add also to your atlas, you know.
That's a good idea. That's a good idea. I hope you're not
So
inspiring
body
reviewers right now, because I think Micha has done that at some time because he's been using the what is it called again? The one that goes to thiols, malimide, right?
Yeah.
So you also use the malimide labeling. Then you can also play a little bit with the pH if you have terminal amines or internal amines to be labeled. So that would definitely be interesting. Thank you.
So I think there's a fair chance that your reviewers will be in this room. So
Well, yeah.
any other questions?
So I wouldn't dare to ask this question in a different environment and to a different presenter. But I'll change it today because Helge and I go back very, very long. So come on, honestly, other than the pan expansion, how much of the Septin work and the Septin tracking could not have been done simply by STED microscopy? Or SMLM if you wanted, but STED as a push-button technique, I mean, it's just easier. So, to align the tubulin everything, how much could you have done by conventional optics based only, and how much do you really need expansion for that?
So that's a hard disagree on that one, because just of the time you need to take the whole image in STED microscopy. So it's a lot of stuff. So that's why we want to team up with people who can do automated STED microscopy. So if you have an automated STED, this is definitely what we're planning right now.
I don't, but Ilaria has, so you
Exactly,
can talk to her later. Yeah,
so that's why I want to talk to Ilaria and maybe
Yeah,
Stefan, because this is what we need to do. I think still the way to go is to combine both, to do expansion STED, because then you can really go to the ultrastructure. So this is now the proof of principle that you can actually do it by using feature-rich staining and principal component analysis to go there.
Why I'm asking, I mean, you know why I'm asking. It's basically because people like, I believe, Jonas Ries, Christian Merrifield, I mean, they did use confocal or SMLM or other type of microscopy to do a somewhat similar analysis for the Clathrin process. But they are they are much smaller objects and they are much more frequent in a cell. So
exactly.
that indeed, I mean, I get this argument absolutely. So you see, you guys, one more application for expansion, semi-large objects that are few in a cell.
Exactly, So these are highly symmetric, very tiny, so you can get 100 in one image. We here have to have one at a time, and it's also huge. This is 10 micron long. You can see that here.
Cool. Thanks a lot.
Cheers.
But now maybe you know one reviewer to exclude, so that's all.
No. No worries.
Okay, so we're doing good on time.
Thank you.
Thank you so much, Helge. And we'll now continue, and as a chair, I'll ensure that we stay on time, although you can get an extra couple of minutes. So we'll continue with Professor Joshua Vaughan, and he's getting set up. So while they connect the computer, I'll just briefly introduce Josh. He's a professor of chemistry at the University of Washington. He did his PhD at MIT, then a postdoc at Harvard, and he has been at the University of Washington for some time. And he's going to tell us about imaging, reconstruction, and spatial analysis of mouse renal glomeruli via expansion microscopy.
I have no idea how to use this. Okay.
Okay, Joshua, thank you.
All right, I'm figuring out the technology. Give me 1 second here. All right, I guess I'm all plugged in from all sides here. Okay, it's a pleasure to be here. Thanks to the organizers for inviting me. Thanks to all the people who worked hard to make this wonderful meeting. Thanks to Helge for leaving me 5 or 10 extra minutes. That's amazing. That never happens. What a 2? What a great guy. So yeah, I'm going to tell you about work we've been doing over the last few years in my group, but I'm going to really focus on this story for the maybe 2/3 of my talk on studying mouse kidney. Before that, though, a personal story. I was on the way to Göttingen yesterday, or 2 days ago, like many of you guys. I missed the train. I don't know, did anybody else miss the train? It was like supposed to be on track 6. It showed up on track 5, and I'm running over trying to get it. I missed it. So I had a little time to reflect on the 10 years that I've been doing expansion microscopy in my group. You know, it's the 10-year anniversary of the first paper. We saw Ed Boyden's great, amazing paper, 2015, and my group was so excited about it, we said we have to try it, but we were also a little bit skeptical. We thought, maybe it's too good to be true, but it wasn't, and we've been really enjoying working with it. So thanks to Ed and Paul and Faye as well. So I just have like a snapshot or two of some of our older work, just to mention a little bit about it. We started off using the DNA antibodies, like many in the early days of expansion. It was working, but we had some challenges with it. So we decided to improve the linking chemistry, for at least to make it compatible with our standard antibodies that we're better at using, and fluorescent proteins. So we did that. We were able to publish that in Nature Methods. Around the same time as a nice story from about ProEXM from Ed Boyden's group, and a related story from Kwan-Hung Chung's lab. Approximately the same thing as ProEXM. So you could see even some of these dividing cell images have now gone into textbooks, which is really gratifying. A second story I just mentioned really briefly was that we decided to find something that had even cooler tubulin than the dividing cell here with the mitotic spindle, and that's Giardia lamblia. So Giardia is a pathogen found in contaminated water. It's actively infecting probably hundreds of millions of people, especially in poor nations without access to clean water. And also it is the master of tubulin. Look at this. It's got this amazing conical spiral of tubulin in green. It's the green channel. And you can see this array is spaced about 55 nanometers apart. So with a mixture of 4x expansion and 2x boost in resolution by structured illumination, we're able to resolve these features very nicely. And we're also imaging 2 channels here. In this case, we have this other protein, delta giardin. So we looked in the literature. We saw that delta giardin was also known to exist on the disk somewhere. People didn't know exactly its exact location. There's, at the time, some just published cryotomography data. And you can see here the microtubules, by virtue of their shape and size, so 25 nanometers across, and they're hollow. And there's also these protrusions that poke upwards, so it's called microribbons. More than that, though, in the same tomography study and in earlier TEM studies, it's known that there are proteins, in addition to the microribbons, there's proteins that localize between the microribbons, and there's also proteins that localize to the base of the microtubules. So we sort of wondered which of these 3 it could be. In fact, it was the microribbons. By looking at the other channel in our expansion sim data, we could see that the delta giardin was directly above the tubulin in each case. So we were able to assign these features from electron microscopy to this other protein that hadn't yet been done. We did also a nice study of a second protein in Giardia, which has got a much less appealing name. It's 86676 or something like that. It's a bunch of numbers. So for neglected organisms like this, they don't have as great names as some of the more trod organisms. In any event, I'm going to skip that part. I'm going to move on. So I'm going to be focusing on the use of expansion microscopy for the study of kidney. We're not the first to do this. We heard great work from Lian Zhao in Ed Boyden's lab at the time, who did expansion pathology, studying human biopsy specimens. It's really great work. I'm sorry the lights are making it a little harder to see some of these things, but you can see here some wonderful work from Hans Blom together with David Unerz-Joyes, looking at kidney here, and you can see these are the slit diaphragms between cells on the filtration apparatus. Here's some of our work a little bit later. So I'm going to get into that. And I guess since I'm spending enough time on this, I need to introduce the kidney in a little more detail. Some of you guys spent time thinking about the kidney. Other of you guys, not so much. So I figured it would be worth going through for a minute or two. So just a reminder, it's a bean-shaped organ. Here's a cartoon of the kidney. It's a cartoon of a human kidney. It's got about 1 million filtration units that are operating in parallel. They're called nephrons. Blood comes into a glomerulus and comes out, and the waste emerges, passes through a long renal tubule where the waste is concentrated and further processed, and then the waste is all combined and urinated out. In addition to filtration, the kidney is very important because it sets the electrolyte levels, it sets the pH of the blood, electrolyte and pH of the blood, as well as blood pressure and a host of other functions. So it's not only important, but actually problems with the kidney are very prevalent. So in fact, in the US, it's maybe 1 in 7 adults has problems, perhaps a little higher than the rest of the world, but not by that much. And it's even worse when you consider that many people with kidney disease don't realize they have it. In fact, I'd say most people don't realize they have it. So it's a prevalent, the problems with the kidney are prevalent and very expensive. So I'm going to be focusing on the glomeruli, renal glomeruli. I think it took me about a month before I could say that without tripping on it. So how does it work? Blood comes in through the afferent arteriole. It splits into a series of, or a network of capillaries. You can see some of the capillary loops here. Waste, small molecules, and some smaller proteins leak out, and they go into toward the renal tubule. This whole structure is encased by something called the Bowman's capsule here on the outside. That's lined by these parietal epithelial cells. You can also see at the core of the glomerulus is the mesangium, so there's mesangial cells there. Zooming into this glomerulus filtration barrier, we've got endothelial cells on the blood side, we've got the glomerulus basement membrane, and then we've got podocyte cells. Podocyte cells are really cool cells that wrap around the blood vessels, and they form these sort of tight stripy features that contribute to the filtration. The endothelial cells have a bunch of little holes in it, so it's like a little screen, and together that's what works the magic of filtration. So traditionally for kidney imaging, it's been studied by thin sections, like a lot of microscopy, and the focus has been on, for light microscopy, and overall physiology and molecular distributions, for instance, immune complexes. Also in electron microscopy, it's generally used for studying ultrastructure. The use of thin sections primarily has caused lots of information to be determined on different sections. So, one thing is studied by EM, another thing is studied by light microscopy. We've had decades to integrate this information, but it does lead to sort of a little bit of a fractured view. So there's, of course, motivation to getting into more volumetric imaging, especially at high spatial resolution. A few years ago, with collaborators, I started to get interested into studying perturbations to foot processes or the thickening of basement membrane. So here's a little patch of a mouse glomerulus. You can see the basement membrane in purple has gotten thickened. There's these foot processes here that have gotten sort of blimped out in aging. Here's another example. This is actually a lineage marked parietal epithelial cells. I'm just going to go back. These guys actually start out here, lining the Bowman's capsule, and it's lineage marked transgenically. But after an injury, these PEC cells, the parietal epithelial cells, can actually migrate inwards and can differentiate into podocytes. And so we can actually see the YFP signal here, or the yellow signals from the fluorescent protein, and we can see it's beginning to form these foot processes as it's trying to come to the rescue of the ailing podocytes. Also, together with collaborators, we started working on doing some fluid dynamics studies of the blood flow, and also trying to come up with more realistic models for glomeruli. So this is one for bioprinting. So this is one from my collaborator. They come up with something really basic that the blood comes in, it splits a little and reconnects. And the goal with the bioprinting is to eventually develop a platform for studying drugs that could be high throughput, and also as a probe for biology. But these sorts of designs were all very artificial because that could be conceptualized, and there wasn't data on for instance, the detailed three-dimensional blood configuration. So a few years ago, my group set out with the goal to create a method to analyze whole glomeruli with sufficient resolution to distinguish all the major components. To do this, we're going to need some good labeling techniques. So we turn to small molecules. So, and some work we had done starting in 2019 with a small paper and later published in 2020. So the idea was to use DNA labels, amine labels, and carbohydrate labels. We've known since the first expansion paper in 2015 that these small molecule labels for DNA work really nicely, which is great. This NH ester labeling, we've heard a lot about it, including Helge Ewers' talk, and I think 4 or 5 talks yesterday. Probably we'll get some more coming forward. Some people call this the PAN label. I think that's a kind of a great choice of name, and Jörg showed this wonderfully in his 2020 paper, how much detail you can get, especially at high expansion. I'd say, you know, even 4x expansion can be pretty good, although more expansion also sounds nice. But in our case, we're getting additional chemical information. And, you know, if you think about PAN, it's, again, a wonderful name. I have no objection to it, but it does sound like you're getting everything. I'd say it's more like getting many things, but it's not getting everything. You can see lots of structures here in green. These are lysosomes. So in Helge's very nice work, he was missing the lysosomes. These were evident in his X-ray data, but not in his PAN stain. So the PAN stain is good for labeling some things, and it's not good for labeling other things. And I would say, you know, these NHS ester labelings have some mysteries that still remain unsolved. For instance, if you guys think what is the most abundant protein of the cell, think in your head, actin for most cells, right, intracellularly. Do we see big bundles of actin in these PAN stains? I don't think we do, actually. So that's just a kind of a small caveat to thinking about, we actually have a lot of bias in terms of what we label. So I generally just call these amines or NHS ester labeling, and it's not a diss to Jörg's wonderful work with the PAN stain that many people call it that. I should mention the carbohydrate stain since nobody mentioned it yet. So here's an example of showing a carbohydrate, a sugar. We've got 2 alcohols. The way that works is we use an oxidant, sodium periodate, to oxidize the alcohols to aldehydes. And then the aldehydes can be uniquely reacted with hydroxides or hydroxylamines to couple them. And these are commercially available. All the probes are commercially available. And so we're able to do this nice detailed imaging. This is just of a cultured cell. I'm going to show you in a minute a whole bunch of data on kidney. First, though, we found that it's a very nice general stain, even for unexpanded specimens. So it's a nice general fluorescent stain. So here we have a collection of, I think it's 30 or 40 different human tissues in this tissue microarray. You can see a couple of them here, but you know, there's a whole bunch of stuff, heart and brain and pancreas. Back to kidney, in cleared kidney tissues, this was imaged by light sheet and it's not expanded. We can see we get quite a lot of detail. Even just the carb channel is enough that we can actually follow these little loops. These are the nephrons. You see these little cross sections through the data in the light sheet data. These are sufficient to trace out these nephrons over long distances. This is over about 1 centimeter of distance. So we can actually map this out just from the one channel, the carbohydrate channel. And of course we've been able to do this at pretty high throughput, but I'll have to save that story for another day. I'm going to focus here on glomerulus mapping. We came up with this acronym GLOMAP. Our collaborators, our nephrology collaborators said, you keep using the word our pipeline or something like that, like these phrases, you need a name. So we came up with the GLOMAP. What are we doing? We're taking mouse kidney sections. We're doing 4x map gels. And then we're doing FLAIR. So the fluorescent labeling of abundant reactive entities was our moniker for that DNA, amine, and carb stain. We expand, we do confocal, and then a bunch of analysis. So we're not doing the biggest gels, we're not doing the biggest expansion, we're not doing the most molecules. We're really doing most of the work on the analysis. So it's really not fancy like a lot of the really exciting, fancy stuff we heard yesterday. And I should mention this is wonderful student who just got his PhD and he's looking for jobs. So if you guys have jobs, reach out. He's ready. So let me show you what some of his data look like. So we've got carbohydrate channel in green, we've got the amines in red, and we've got DNA in blue. Okay, so I'm just going to acquaint you with this data set and let you get your bearing. So you can see here the Bowman's capsule goes around the glomerulus, marking what's called the renal corpuscle technically. We can see this inner membrane is the glomerulus basement membrane. That's where the filtration occurs. And if I can figure out where we are. So here is right here is the incoming blood. This is the afferent arteriole. It comes in towards this vascular chamber, and you can see it splits off into a bunch of different capillaries, so a little network of capillaries. These wind their way around the glomerulus, and they're eventually going to escape here. They're all collected into the efferent arteriole here and move on their way. Okay, so you can see the blood goes in, blood comes out, and then we've also got the proximal tube here, where the waste is exiting the glomerulus. Okay, great. So my poor student Adil worked through manual segmentation for several glomeruli. This was extremely painful. I'd say each one took him a few weeks. Okay, so he was struggling away with this, and we had a game plan. But first of all, you can see these wonderful hand-drawn segmentations going through all the planes, labeling everything. We tried to use some computational tools at the time. It was partly working, partly not. So he just rolled up his sleeves and did it, which was very impressive. But the plan was that we would use those first 4 or 5 hand-annotated sets as training for a machine learning model. So we got help from our University of Washington colleague, Professor Sheng Wang and his lab, on setting up a 3D neural network to use those 5 initial hand-segmented glomeruli in order to create a machine learning model that could do most of the heavy lifting. And so in a couple of hours, we could then get rough segmentations out. These could get refined in about 2 days per glomerulus, and then a little bit more post-processing, fiddling, clicking buttons, and so forth. So it took about 2.5 to 3 days of human time, which was a big improvement. I would say it's still a pain in the ass, but it does pretty well. It's enough that we can start to learn quite a bit. How's the segmentations? Just to show it. So this is a set. This is the computer analyzed and then hand edited. So it's just refined and sort of double checked. You can see the nuclei. You can see the basement membrane here in blue. The blood space, the mesangium, the Bowman space is the urine space. So we can actually map out all these structures really nicely. Here's the zoo of 24 glomeruli that he studied for a different age range. We call these adult, 2 to 20 month mice. We've got 27 to 29 month mice, so very old mice. All of them will have impaired kidney function. And then we've got a disease model, which I'll tell a little bit more about later. So beyond just identifying the key structures, Adil was able to determine all of the cell types. This is a little bit of a challenge traditionally to do certainly all at the same time with studying glomeruli. First of all, sometimes the nuclei are very close together, and it might not be easy to resolve them. Second, because of that resolution challenge, it has traditionally required the use of antibodies to prove that you're looking at the cell that you're interested in, rather than just taking a guess. But with the spatial resolution we had, we were able to determine the identity of each of the cells by virtue of the nuclear shape and its position. So for instance, this nucleus, let's see, which one do I want to pick? Yeah, like this nucleus here is part of the mesangium. It's sort of more inwards. It's inside of the blood. It has a bunch of carbohydrates channel outside of it. This guy is an endothelial cell. It's in a capillary loop here, and it's just towards the edge of it, so it's in contact with the blood. We've got another cell here as a podocyte. Here's the nucleus, and it's just outside the basement membrane. And then we've got the PEC cell here lining the Bowman's capsule. So we can map out every individual nucleus and count all the different cell types. We did the antibody tests in order to validate this, and we found we had about a 95% accuracy. Okay, so I'm going to show you a visualization of the nuclear model here. So I'm going to go through this movie. We'll see if it plays too fast or not. That was too fast. Let's try that again. Okay, so here you can see the PEC cells lining the Bowman's capsule. You can see the podocyte cell or cell nuclei just outside the basement membrane, filtration. See the endothelial cells sort of mixed in with the blood. Again, the nuclei mixed in, and we can see the mesangial nuclei within the mesangium. So we get a very nice view of exactly where each cell nucleus is located within the glomeruli. Tons of data distilled down to a small table. You know, in a way it's awesome, in a way it makes me cry to think about how many, probably a terabyte or more of data just comes into this, you know, like 25 numbers. But yeah, so here we've got the 3 groups, adult, aged, and FSGS, and we've got the cell counts for the 4 principal cell types. Interestingly, we got higher cell counts than were known in the literature. We think that it partly has to do with the fact that we had higher spatial resolution and could resolve these tightly, sometimes very close together nuclei that would easily be confused for a single nucleus. But probably also our limited sampling could have biased us towards glomeruli that might have been bigger or had more nuclei. So that's something where we need a little more work to really prove that. What else did we see? We saw that the endothelial and the mesangial cells numbers within the age population grew a lot. That's sort of well known. It's called a mesangial expansion in age. In the FSGS model, which is a disease model for mouse kidney, which causes an injury to podocytes, we see indeed the podocytes are depleted, and these PEC cells, parietal epithelial cells, that try to come to the rescue of the injured podocytes, they're actually upregulated. So we can see some really nice distributions and get the absolute counts in nuclei, which is really not something people have had for absolute counts of nuclei in whole glomeruli before, certainly for all of them together. We also found interesting shape metrics. There were actually many other tables like this with lots of interesting phenotypes, but a simple one to explain is just that all the nuclear volumes were increased. So this FSGS model, it's a focal segmental glomerulosclerosis. So focal means some of the glomeruli are injured. Segmental means within an injured glomerulus, which is the only kind we studied here. You have sometimes a more diseased or distorted looking region and a mostly healthy looking region. In this case, we've taken the glomerulus and we kind of cracked it open like a walnut, and you can see the healthy side on the left and the distorted side on the right. The distorted side has a kind of a swollen mesangium, and you can see that correlates to a reduced blood volume that's sort of got a bunch of dead ends, and so half the glomerulus is just not really working properly. We can see the podocytes have been primarily injured on the distorted side of the cell, and if we look at the PEC cells and we map them out, or the PEC nuclei, we can actually see if we map out the distance of the nuclei to this basement membrane, we can actually see larger distance from these PEC nuclei to the basement membrane on the distorted side than we can on the healthier looking side. And so the heat map shows more blues and greens here on the right, and mostly reds, a couple yellows on the left. Okay, so we can see a snapshot of these PEC cells coming into well, also proliferating, but also coming into to try and pick up the slack and help out the suffering podocytes. Okay, the last little bit. So maybe I have 3 slides left. So we did some analysis of the basement membranes. This is work done by a talented undergrad on this project. And so we had these nice segmented models, which we used to create these 3D maps of the thickness of the different basement membranes. So here's the Bowman's capsule. We analyzed all the glomeruli from our study in this way. We could see the aged Bowman's capsule basement membrane is much thicker, right? So aged is this sort of blue, right? And in the histogram of thickness, we see the blue tail extending much further to the right compared to the adult one. If you look at the color map, you can see this large patch of thickened, kind of almost sclerotic basement membrane, and it's right next to the proximal tubule junction. This is a known site of injury in aging in kidneys, so to see these injuries to the proximal tubules, and we found this in all 3 of the aged BCBMs from the aged glomeruli that we study. In contrast, this model disease, which was for approximately 3.5-month-old mice, but that had been subject to an antibody injection that attacks the podocytes. So in this case, you can see one half of the glomerulus not necessarily next to the proximal tubule. One half of the glomerulus has this thickened basement membrane and the other did not. We did the same analysis on the, so that was Bowman capsule. We did the same analysis on the glomerulus filtration basement membrane. We see thickening in aging, but the thickening is more diffuse, right? And so we can get out a bunch of novel phenotypes about using our GlowMap assay, not only monitoring the thickening, but also things like the total surface area of the basement membrane. This isn't something one could easily get in any other way, and now we've got access to these novel phenotypes. So I told you what we can do with it. All of these raw data, the segmentations and the 3D models that can be sort of interacted with, they're all online and downloadable. We've got hope that with a little bit extra training on our full set of 24 models that we could do a next generation with faster and analysis, less human time. We're interested in taking this further by correlating injuries to the glomeruli and tubules. It's a well-known phenomenon, but has not been easily accessed before. And then we're working together with collaborators on some computational modeling and providing accurate digital models of the blood volumes for bioprinting efforts. So big thanks to my talented graduate student or former graduate student, just graduated, Adil. Again, he's looking for a postdoc or a job. And thanks to many other collaborators who worked on this project and the related projects, and of course to the funders, and I'd be happy to take a couple of questions.
Thank you, Joshua. So we have time for maybe 1 or 2 questions.
Maybe I missed it, but which type of microscopy did you use here? Because you are really looking at something big, right? So in terms of working distance, I guess you have to go for.
Yeah, we're just doing point scan confocal. So we had the tissue sections are about 150 micrometers at the thickest. 4x expanded gets us about 600 microns. So we had a I'm trying to remember the NA, 1.15 maybe, or 1.2 NA water immersion lens on a point scan confocal. The imaging took about 90 minutes. We would image 3 channels in parallel and did a little bit of unmixing.
And you didn't see any issue of signal at deeper?
We didn't get a lot of bleaching. Flare is very bright.
Yeah, okay.
So there was no special corrections for depth or anything. I mean, you saw the fly through where I went through, yeah, and there was no
correction.
Yeah.
Okay, thank you so much, Joshua. If you have questions, please reach out during the coffee break, and I think we stop on time.
Such a nice meeting. I'm really looking forward to interact with all of you. So indeed, my lab is focusing on morphogenesis. We try to understand how stem cells are interacting with each other to build organs. And we have a fundamental question, which is to try to understand how mechanical forces participate in the process by regulating cell identity and cell shape. And we have a focus on cardiovascular development. The reason for this is obviously the cardiovascular system is really important to just sustain our function, our life, and it's involved in a number of pathologies. And also, in terms of development, the heart is the first functional organ in our body. And it's going to function, meaning that it's going to beat and move blood in the body before it's completely mature. So this creates a challenge for the tissue as it grows, because there will be some additional mechanical constraint that the cells will have to deal with. And we know that these mechanical forces are really important for the proper growth of the heart and its maturation. And if they are abnormal, they will lead to pathologies that are known as congenital cardiac diseases, which can be really detrimental. So to study mechanical forces in vivo, we use an animal model, which is the zebrafish that you can see here, looking at us. And an amazing feature of the zebrafish is that you can keep it transparent for a while, and you can image it at high temporal and spatial resolution in time. So, for example, we can really depict very carefully the mechanical forces that are generated, for example, by the flowing blood cells, generating shear stress on the surface of the cell, and by the contractility of the cardiomyocytes that are forming the myocardium, which are the active part of the heart that will allow the heart to pump. Again, these cardiomyocytes are functional very early. So to get to a mature heart that you have a sort of a picture here, it's still very embryonic, but after 3 to 5 days of development, you will have a heart with all the cell layers that are found in mammals as well, and which underlie the function of the heart. The anatomy of the heart in zebrafish is simpler. There are just 2 cavities, a ventricle and an atrium. And for our work, we mainly focus on this area of the heart, which will contain the cardiac valves. This area is called the atrioventricular canal that separates the ventricle from the atrium. And the cells that will shape and build up the cardiac valve are called endocardial cells. So they are specific endothelial cells of the heart. And there are a number of pathologies associated with valve developmental defect. And we know from numerous work from our lab, but many others, that the flowing blood that you see here in the atrioventricular canal is really important to identify the cells that will form the valve. And if there is no mechanical forces, valve will not develop properly. They will be actually completely absent. So for us, it's a very nice system to integrate the mechanical forces that we can record at high resolution, and also the cell behavior and cell identity that we can also image pretty well. So today I will just focus on an early stage of heart development, which is really important, which is called the heart looping, where you have a very big morphogenetic process which is happening, which is actually allowing to establish the 2 cardiac chambers, the ventricle and the atrium, from this linear heart tube. So this is how all hearts develop first. They assemble as a linear tube, which you can beat, and throughout the looping process, you will have the morphogenesis of the heart and the separation of the cardiac chamber. In that case, it's just going to be 2 cardiac chambers. Importantly, you see that this cardiac looping is associated with the formation of this structure, which is the atrioventricular canal, where the valve will come out. These cells, again, are very sensitive to mechanical forces. And using different type of imaging, we could segment the cell shape changes associated with the morphogenesis of the atrioventricular canal. And we could find that initially in the linear heart tube, the cells are have a fairly consistent shape, but through the process of morphogenesis, specifically the cells of the AV canal will become much smaller, and this will allow the morphogenesis of the ventricle and the atrium. So the whole structure depends on each other so that you can shape properly those cavities. So we can measure cell size, we can also measure cell volume, and we found that the cells specifically in the AV canal are actually shrinking in all dimensions, so they just become much smaller during the process of looping, generating some change in the mechanical properties of the heart, and also most likely helping the morphogenesis of the ventricle and the atrium. So this area of the AVC is very specific because it also is in contact with higher shear stress. And we found that these cells, again specifically in the atrioventricular canal, have a unique calcium pulse pattern, which is mediated by mechanical forces. So you can see this is light sheet microscopy of a GCAMP7 specifically expressed in the endocardium. So this is not the myocardial cells, it's really the cells that line the heart that are in direct contact with the blood. And the black cells are the cells that turn on these calcium pulses. So there is a very specific set of response of these cells, and we could find that if you stop the heart or change blood viscosity, these calcium pulses would change. I have to summarize a few years of work in that slide just to tell you the process of this cell volume change. We think it's really important to understand it because it's really at the basis of the looping of the heart and the establishment of these cardiac valve cells. And we found that this change of cell volume is mediated by activation of piezo, which will lead to calcium fluxes in the shape of these pulses I showed in the previous slide. And then some signaling will occur so that the cells will change their porosity to water and they will shrink. So we think that there are a number of events that lead to this change of cell volume, which are activation of mechanotransduction through the stretch-sensitive channel piezo, but also change in osmolarity of the cells so that they can adapt their volume and just shrink. So there is a combination of pathways that are involved, but also at the morphogenetic level, there is a very interesting feature, which is the fact that by shrinking, the cells of the AVC are actually becoming much smaller, and their nucleus follows this trend. So if you measure the nuclear volume, we found that it's much smaller in the AVC by comparison to the rest of the cell, and it scales with the cell volume itself. So there are some big changes in the size of the nucleus, and we wonder whether these changes are associated with change in cell identity. So the nucleus is a very complex structure, and DNA interacts with its periphery through different types of protein that will connect to the bilayer of the nucleus. And it will interact through some sets of protein that forms the nuclear lamina that is lining the inner part of the nucleus. And it's known that DNA packed with chromatin has a tendency to be silenced when it's at the periphery of the nucleus, when it interacts with the nuclear lamina. The nucleus is also mechanically coupled with the rest of the cells through the link complex. And overall, it's really actively dependent on the mechanical forces that the cells are experiencing. So we are very interested to try to find out how chromatin is impacted by mechanical cues that are generated by the contractions, but also by the change of osmotic forces that are associated with the water fluxes activated by aquaporin that we show is important to change the nuclear volume. And there are a number of elements that show that nuclear function and nuclear mechanics is associated with change in osmotic forces and mechanical forces. So people have shown that if you change osmotic strength in the medium, cells will tend to change their chromatin compaction. Their chromatin organization is also variable depending on the strain cells will experience, and they will also change their nucleus in response to these mechanical changes. So we thought that our system could be a nice model to pinpoint more precisely the interaction between mechanical forces and nuclear mechanics and organization, and also assess its impact in the process of cardiac valve morphogenesis. So the first thing we did was to assess chromatin distribution in the endocardial cells using some basic immunofluorescence targeting histone marks that are known. So I show you here H3K27 acetyl. So the contrast is not great, but we have a staining here for the nuclei with DAPI. This is the labeling for H3K27 acetyl, and all of these dots are the nuclei, so the staining is very specific. And we have a counter stain of the endocardium, so you have to trust me, but we can separate endocardial cells from any other cells because this H3K27 acetyl mark is present in all of the cells of the body. So what we do is to segment the signal using the cathedral channel, so we are sure that we measure intensities in the endocardial cells. And we segment the nuclei, we then identify their position, and then we can extract the fluorescence intensity in each of these compartments, ventricle, AVC and atrium, and we can quantify whether there are some differences in terms of histomodification. So this is how typically people do in cell culture, and they found that mechanical stress actually alter specifically H3K9 trimethyl. And interestingly, so when we plot these measurements, so these are normalized measurements with the intensity of the endocardial channel, we can see that depending on the cell type, we have some different intensity of the chromatin, which of this chromatin mark, which suggests that there are things that are very specific that are happening in particular in the atrioventricular canal. So we thought that it's very interesting. And however, as you saw, these are confocal microscopy image. You cannot really see their localization inside the nucleus, obviously, because the resolution is not good enough. So obviously, we turn to expansion microscopy. We think that these scales are very much adequate, so the nuclear scale are very much adequate for expansion microscopy. I suspect that you know all of this. There are different families of expansion microscopy, EXM and MAP. And recently, some very nice work from the Giraldes group showed that you can actually use expansion microscopy to assess in high detail chromatin organization and transcriptional activity. So we definitely focused on the method. So initially, our work has been very much influenced by the work of Virginie Hamel and Paul Guichard, who developed UEXM. And the reason for this is mainly because Emmanuel Stab joined my lab to develop expansion microscopy, and she spent her PhD with Virginie and Paul, so she had a huge, a good feel about how to develop such a method. And, well, basically, she termed it tissue XM. Her idea was to keep the integrity of the entire animal, which is small enough. We're in the order of hundreds of microns, so that she could treat the entire embryo through an expansion protocol and then analyze it in the best way possible. So one of the huge advantage of the method, I mean, so the protocol is very similar to the UXM from Paul and Virginie. There are some changes, in particular in the length of the treatments because of the size of the tissue. But so it makes the method last for something like 2 weeks. But it's really worth the wait. One of the major advantages is that you can scan through the entire animal. So this is a larva, it's not an adult, obviously, but this is what we do. So the localization and the orientation in the animal is really easy. And then you can simply focus on one of the areas, zoom in, and perform high-resolution imaging of the ultrastructure of interest. So it's a huge benefit by comparison to other methods that would allow us to do super resolution. They are much more difficult to implement between electron microscopy or super resolution approaches. The gain of time is amazing. So a few words about the validation of the approach. Emmanuel did a number of tests. One of them was to assess for the length and structure, I mean the diameter of the basal body and the primary and cilia, which is a nice way, a nice internal ruler to assess for the expansion isotropy. And she could see that there is no changes between the tissue observed, very consistent expansion throughout the animal. And the gain of resolution is amazing. I mean, so for example, here we focus on one of the motile cilia of the lateral line, and you can see the microtubule doublets and the central pair of the motile cilia. You can visualize here the membrane, so the ciliary membrane, with a very an amazing resolution. I think we never saw that before. And the huge advantage is that you can use a very basic confocal microscope, providing that you have the proper objective at the proper magnification and NA. So it's not only adapted for zebrafish. We made some experiments with mouse embryo as well, and we could expand those very beautiful cilia in the node of the embryo, which is an important structure to set the left-right axis of the embryo. And overall, we think we have a very nice method to assess ultrastructure within complex tissues. So to go back to the nuclear studies we'd like to implement, well, basically what we did was to use tissue XM and focus our analysis on the nuclei. And this is the work of Alexia Pigeot in the lab, who started her postdoc recently, and it took us some time to set the method up for the nuclear modification, but the results are really encouraging. So these are like very much ongoing work. It's the beginning, but I'm really excited to share these images with you. So basically we are visualizing here the heart. This is a DAB staining. This is a labeling of the endocardial cells. And if we zoom in the atrioventricular canal, you can see the nuclei of the endocardial cells next to some nuclei of the myocardial cells. And we have this H3K9 trimethyl, so histone modification labeling. And you see this is amazingly beautiful. So from this quantification we would do with a regular confocal microscopy, where you could just analyze intensities. Here you see distribution of these, or accumulation of this histone mark between the different cells. You can also see the change of chromatin compaction using the DAPI staining. We can look at different cell types. We have different cell lines also that will label different aspects of the compaction of the nuclei. And amazingly, you can see these changes in chromatin distribution depending on the type of cells you are looking at. It looks like the myocardial cells accumulate this H3K9 modification in the periphery of the nuclei, whereas it's more even in the endocardial cells. All right, so work in progress. The conclusion is that tissue XM allows us to visualize the nuclear deformation, the chromatin compaction, and the chromatin organization in the endocardial cells. We have lots of ideas, obviously, for the future. We'd like to label chromatin modification in vivo, go out from antibodies. We'd like to assess mRNA localization. There are lots of work for improved mounting strategies, and I'd be happy to discuss this with you. And also we're working with Marine Laporte, who is here, on developing alternative fixation strategies to keep the native state of the structures. All right, so big thank to the lab, Alexia and Emily, who are here, Marine, our collaborator, and you for your attention.
Thank you very much. Do we have time for maybe one, maybe two questions, if there is an audience, some questions? Then we move directly to Utrecht.
Hi, those really nice visuals, thanks. My question would be, because now you work with the whole organism, how do you tackle the acquisition? So you mentioned you go from the full organism to the specific location. Do you do it within one imaging session? And if yes, how long is it?
Yes, so it really depends on the question. You don't need the entire animal. Once we focus, I mean, we focus on the heart, so we really keep the whole embryo for showing off in a way. But I mean, it's really important for us to orient ourselves in the tissue. But then for the imaging, we can just focus on the area of interest. I mean, there are some issues with depth. So the type of objective we use is critical, because with the expansion, the animal becomes big, and we have to deal with it. So we also have approaches to extract the heart and just get the heart on the slide. So we limit the issue with the depth, and the resolution is amazing. But overall, if you need to keep the entire animal and image it all, I mean, it takes quite a while. So we do tiling, and yes, you need to have the confocal time for that.
Okay, then thank you very much, Julien. Again, another. Okay. Now we have the pleasure to have Ute Becherer. She also studied in Strasbourg, but did not developmental biology. She did more biology and neurosciences as a PhD. Then also moved to the US. In this side is Colorado Denver University, to then actually spend some time here in Göttingen, in the Max Planck Institute. And in 2001, she found a place in Saarland, in the University of Saarland, where she is until now. So I guess Ute will basically explain or show us some deep learning or adaptable deep learning platform for automatic detection. Thank you very much.
Before Uta starts, I promised her to embarrass her on stage. So Uta is my mother doctor, and she's great mentor, and she holds a special place in my heart. So thank you for coming, Uta. It's always a pleasure to see you and connect with you, and I'm looking forward to your talk. And sorry, Philippe, for crashing your session.
Well, thank you for inviting me. It's sort of a homecoming because I spent 2 years here in Göttingen. And so it's really nice to be here. My talk will be on a subject that is quite far away from expansion microscopy, but it could be a platform that might be useful for super resolution, so D-storm and any of these sort of methods. This program, the IV program, has been devised entirely by Abed Shuaib, who has his poster outside, who's my PhD student, pretty much at the end of the PhD, looking for a postdoc. He did a fantastic work to actually automatically detect exocytosis event, but this program can be used for detecting any sort of activity in fluorescent videos. So what was the challenge? The challenge was to detect automatically exocytosis in diverse type of cells. And so you have 3 main type of measurements that you can have, either in cells like these, where exocytosis is visualized from individual vesicles that are moving all over the cells and can fuse on the membrane anywhere on the cells. So this would be one possibility here. The other possibility would be to detect exocytosis of synaptic vesicles in neurons, and there the exocytosis occurs always at the same spots, meaning the synapse. The vesicles are very small, so you cannot really resolve them, and when you have multiple exocytosis events one after the other, what you see is a slow raise in fluorescence intensity and slow decay of fluorescence intensity. And then the final way to measure this is to do nanosensors, where you measure here the release of the neurotransmitter, for example, and there you have sort of big areas of fluorescence intensity that increases. So I'm not going to speak at all about this last method. I will speak a little bit about this, but I will mainly concentrate my talk on this because of a lack of time. So here in this case, in these cells, where you see individual vesicle fusing, what you can have is like 4 different type of signature for the fusion. In one case, you labeled either a protein that is in the membrane of the organelle, or you label the protein that is in the cargo, so the one that is released. And you can label it with fluorescent indicators that are pH insensitive. In this case, what you see is merely like a cloud of fluorescence of what is released and just a drop of fluorescence. But you can also label them with something that is pH sensitive, that is quenched when it's in an acid environment and is dequenched when it's released at a neutral pH. And in this case, what you see is a sharp increase of fluorescence prior to exocytosis. So you have a different signature of the fluorescence intensity over time for each of these events. So that means you cannot devise a program that is based on mathematical operation, because you have very different type of signature in the fluorescence intensity over time. So a lot of programs have tried to do this based on mathematical algorithm, but they failed. They've worked for one specific type of event, but not for everything. And Abed tried this as well, and he failed as well, like everyone else. And then he said, okay, let's put this aside and let's go for artificial intelligence. And there he devised two different methods that we use for two different type of exocytosis event. One that we use for the synaptic transmission, where we don't have mobility, so we don't have this problem. And the other that we use for all the other cell types, like neuroendocrine cells, endocrine cells, or cytotoxic T cells, where you have mobility of the vesicle before they fuse. So what is the program doing? First, it recognizes any type of activity in the movie. So basically it detects fluorescent variation, and it looks where this fluorescent variation occurs, extracts this as a region of interest, and it extracts sort of a little video, a sub-video of, say, 25 frames, or more or less 25 frames. And then it analyzes these small sub-videos, either here in this, here it analyzes the fluorescence intensity in sub-regions of these videos, generating these sort of graphs. And out of these graphs, it can identify whether you have fluctuation just because of loss of focus, or movement of the vesicles, or actual exocytosis. And then the neural network selects out of all these graphs the one that is actually exocytosis. The other option, and this works really well when you are not interested in any movement, but if you have movement, then this doesn't work well, and it's not very adaptable. So when you have movement, like in endocrine cells or CTLs, he uses a different method. He always still extracts all these different regions of interest as little movies. Then he encodes all these little movies, and out of this encoder, then we have another neural network based on vision transformers that analyzes the patches themselves, how they look. And this required a lot of training, and out of this training, it then extracts the actual exocytosis event, because they have a certain look. And so then I'm now going to concentrate mainly on this type of analysis. And the first thing that then Abed did was to do some benchmarking. And he did some benchmarking by using a video that he actually made. So it's a virtual exocytosis event that he actually designed, and where he applied different level of noise. And by doing this, he generates movies that look like this: low noise, middle noise, high noise. And then he needed to have some parameters to see how good the network performed. And you have two parameters to analyze this mainly. It's the so-called recall parameter or precision parameter. The recall parameter is a parameter that tells you my program detects everything. So it measures the number of true positive and divides this by the number of true positive and the number of false positive. The false negative. The false negative are those events that are missed, basically, by the program. So this is one of the parameters. The other parameter is the precision. There you see if in the event that you detected, you detect some events that were not real events. So here you divide the number of true positive by the number of true positive plus the number of false positive. So when you analyze these type of movies, you see that when you have very little noise, the program performs superbly. You have 100% recall and precision. When you add a little bit of noise, like in here, this would be representative of what we usually have in our movies, then the program still performs very well. You have a very high recall, close to 100%, but the precision goes a little bit lower. So you have some false positive in what you have as detection. When you add a lot of noise, like here the level of noise is the same level of what you actually want to detect, then the recall goes down and the precision also goes down. But to be very honest, these type of movies we would not even bother to analyze because they're too bad. So then, of course, we needed now to test it on real experiments. And the first experiment, or the first type of movies that he looked at, were movies where the exocytosis events had a lot of feature. Features means the vesicle very clearly visible, so big, large secretory granules. This is the case in cytotoxic T cells, where the lytic granules have a size of about 300 nanometers. And also where the granules are stained with pH sensors, so that you have this clear increase of fluorescence prior to or during exocytosis. So the movies look like this, clear flashes when you have the exocytosis occurring. This is actually fairly easy also for the human expert to see. It takes time. Per movie that was like 10-minute movie, you take about an hour to analyze, to detect those, but the program detects them also very well, but then in 30 seconds or less, just a couple of seconds. And when you then compare what the human expert is doing as seeing as true positive and what the program is seeing, you see that it's the same amount, even the program detects slightly more. It didn't get any false positive, and therefore the recall and the precision is extremely high. Let's now move to a movie where the vesicle exocytosis has a little bit less feature. So we stay in these cytotoxic T cells, so still pretty big vesicles, but this time we use labeling that is not pH sensitive, that has been proven to be very difficult to detect for any other program that is on the market. Because we just have the drop in the fluorescent intensity, and you can see it, or maybe not. Is the movie running? No. You see, basically, you should see in this movie, yeah, there's a purring vesicle, that's all. And the human expert takes time to visualize these events. The program is a whole lot faster and it detects the exact same event plus additional one. Are these true events? In most cases they are. And actually the program is better at detecting weak events than the human expert is. And it has few false positive events. So it detects some additional events, but I'd rather have a program that detects too much that I can sort out quickly than a program that doesn't detect real positive events. So at the end, we still have a very good recall, that is about 98%, but the precision went down a little bit to about 80%. So we have 20% of false positive events. Then we move to completely different signature. Here we're looking at the exocytosis of multivesicular body releasing exosomes that are labeled with pH-sensitive markers. So here, the release of the exosomes, when they're released, they don't separate really quickly. They stay on site for a long time and then slowly move away from the exocytosis site. So what you have is you have this fast raise of the exocytosis with a deprotonation of the pH sensor and then a slow decay of fluorescence. For this, Abbott had to train the model again to see this different signature of the exocytosis. But when he did this, then you see here that the human expert saw these events and the program saw the exact same events. So when he did this over many movies, you can see that the number of true positive is a little bit higher than what the human expert could see, and we have very few false positive, meaning we have a very high recall and precision of the program. How does our program compare to other existing programs? So we tested a couple. Here's shown only the result of one program called ExoJ, because it's the one that performed the best among all of those that we tested. And what you can see, ExoJ performed very well here to detect these type of events, because it was designed to precisely detect that and nothing else. So it did its job. But then when we applied it to other type of events like these, it performed much poorly. You see, this is always these orange bars. They're much, much worse than our program. Then we turn to cells where the vesicles have even less features. So these are endocrine cells where the vesicles are smaller. They're just about 150 nanometer in size, or even smaller than this, sort of like 60 nanometer. So we have chromaffin cells here where we have large dense core vesicles, or we have here beta cells that are filled with insulin, where the vesicles are filled with insulin and the vesicles are much smaller, as you can see here in these movies. And we stain them with fluorescent marker that are pH insensitive or weakly pH sensitive. And here, the vesicles, when they exocytose, they just lose their fluorescence intensity, as you can see in this movie. And the human expert has a hard time to see all the events. It takes a tremendous amount of time, 2 minutes of movie, 1 hour of analysis. And it has all these events, and the program actually could detect more. Are these real events? Yes, they are. They're just very weak. Or because it's so tiresome to analyze this, you just miss some. And so you can see that we have a lot of real true positive events that were missed by the human expert, and you have false positive, but not that many, and they're easy to discard in post-process by the human expert afterwards. So we have very high recall. The precision drops a little bit to 80%. What happens here with the weakly pH-sensitive labels? It also performs really well, the program. Here's the result from the human expert. Here the result from the program. And as you can see here, it performs really, really well. And the recall is high, the precision is high. I'm trying to speed up. So all this works fine. Then we said, does this work with other type of events? Completely something different. So we got from a neighboring lab in cardiomyocytes some measurement of calcium sparks. Can we detect those? So the calcium sparks look like this, very different than what we have. And yes, the program can detect it very well here with the original models, no retraining. But when we retrained it a little bit, we could detect even more. It was just a little bit of retraining. So it works super well. And then I thought, okay, I'm in a super resolution. Where do we have activity? We have this for D-storm, for example. And we had very old D-storm data in our lab that is horrible. Nothing in comparison to all the movies that I've seen here in this audience. So a really horrible movie, but can we detect something in a movie like this? And yes, we can. The program could detect, but was not doing very well. So this is the program as I showed you now. The other line of detection, this other model that we use could detect much better and is actually more effective in this because you have so many events that the other model was like overwhelmed. So we needed something that is able to measure more and quicker. So it can do this. So if you have difficult movie to analyze, you might be able to use this. However, then it has to be paired to one of your program that you use normally to do the precise localization and the reconstruction of the data. Is it easy to use? Yes, it's a plugin in ImageJ. So anyone can use it. You just put it as a plugin in your plugin thing from ImageJ and you can use it. It comes with a complete manual, and the GitHub site, you also have some videos explaining how to use it, and it's really not complex. You don't have many parameters to set. And then, as I said, you can retrain your model. You have a training tool that is coming with it. I'm not going to go into it. And how complex is the retraining? Or if you want to train a completely new model, you can do it, but that takes time. You need about 200 events per category of events. You have to have about 10 different categories. So it means you have to have about 2,000 type of events, or 2,000 events to train on. That takes time. If you just want to retrain an existing model, you have to add like 1 or 2 different categories. There you need only 20 or 30 events, and then there you are. So it's like cell post 2. When you retrain it, it goes super fast. Same here. And then the program comes with a little bit of goodies for any one of you who's doing fluorescence intensity measurement over time. This is the great goodie, because it measures fluorescence intensity in a certain time frame that you can set around the time point where you set your region of interest, and it measures them all at once, and you get graphs like this. And it does this also on multi-channel images, and then you get the graphs as you want to. So this is the one goodie that probably everyone who's using the imagery will need. And that's it. And I'd like to thank the people who contribute to this work. So the main person is, of course, Abbott. His poster is outside is the poster A9. The movies were made mainly in our lab, but we had also movies made by other people in different labs in Sweden, in France, and from people here in Göttingen. We get great support also from Constantin, Pape, who verified that everything was correct in the program. And we were supported by the DFG and the ERC in our Synergy grant attack. Thank you for your attention.
Thank you very much, Ute. Do we have one question that is very pressing? Otherwise, we do it later with Ute. Thank you very much, Ute. We can now move to Constantin. In the interest of time. So Constantin did his PhD in Heidelberg, I think in computational biology or computational science. Yeah, computational science, I believe. Then he spent a very intense year in Janilia farm, came back to Heidelberg, EMBL in Heidelberg for a postdoc, and now we have the luck to have him here in Göttingen, so helping with all our image analysis. It's great to have you here, Constantin. The stage is yours when it's possible.
Okay, perfect. Thanks for the kind introduction. And yeah, as already introduced, right, so what the lab is doing is mainly developing image analysis methods sort of for a variety of applications. For this talk, I want to first introduce maybe some recent developments in XEM, right? So one thing we see here is this whole body of a zebrafish imaged with expansion microscopy. So the preprint on this came out very recently. That's work from Janilia, right? So work we'll hear more about later is expansion microscopy really at the synaptic resolution. So this is this Lycon work where we can do things similar to connectomics, right? So really reconstructing neural circuits, but with XEM. And then, right, we can even go to the nanometer resolution, right? So this is work by Silvio and Ali, I also contributed a bit to, where we can see individual protein shapes, right? So we see that expansion microscopy really works across scales, right? From analyzing kind of the morphology in full animals to synaptic resolution to proteins. What all of this data has in common is it's very large and very complex. So we need advanced deep learning or other analysis tools in order to extract the knowledge that we're interested in from it. And so this is where my group comes in, right, in developing these analysis solutions for this data. Right? So what we do a lot is building kind of these custom segmentation solutions for large and complex data. So these are just two examples from the lab. So the first is SynapseNet, right, which you see here, which can analyze synaptic data in electron microscopy by segmenting vesicles and other structure. Or here, this is CochleaNet, where we can take cochlea, sort of the whole organs imaged in light sheet microscopy, and extract the neural cells, right? So hair cells, spinal ganglion neurons, and so on. And right, so all of this really is very helpful for specific tasks in automating these analyses that would not be possible because this data is so large, but kind of these custom solutions come with its challenges. So the main thing is that building them requires a lot of specifically labeled training data, right? So for example, for SynapseNet, we used around 120,000 vesicles in 350 tomograms, CochleaNet 300,000 cells in 40 small volumes. So providing this training data takes a lot of work, and it only works then in very similar imaging settings to the original training data. So overall, this means that it's a big effort to build such a solution for a new system. So one of the main goals in the lab is to try to build more data efficient methods, or just methods that work kind of without these training for new imaging modalities. So and luckily, the overall computer vision community has not been sitting idle on this, and in particular, there's now this model called Segment Anything, which can segment a lot of different structures in different kind of images. Right? So you see, so from this original publication, all of these different kinds of mostly natural imaging here, where the model can automatically segment structures in. And this was achieved basically by collecting a really giant data set, 11 million images with over a billion annotated objects. And in addition to automatic segmentation, what that method can also do is interactive segmentation, right? So you see here in this GIF, basically there's kind of the cursor as the input to the model, and then the blue thing is a mask that it predicts, and as we see here in this case, this can even be kind of refined with further annotations. So we've seen with this model, which came out about 2 years ago, that more robust, more versatile segmentation that works out of the box in a lot of different settings is possible. And so then we ask ourselves, can we do the same for microscopy? And so the answer is yes. And so what we did is build this model called Segment Anything for microscopy. In short, we call it microsam. It was published beginning of this year in Nature Methods, and where we adapted and improved Segment Anything for microscopy data. So the way we did this mainly is by collecting kind of a large data set of diverse light microscopy and electron microscopy images, sort of from already published data, and then we retrained the Segment Anything model on this. And we also added a new part to the model that improves the automatic segmentation, right? And you can see here for the automatic segmentation, this is the original output by Sam in this phase contrast image of cells. So we see that, I mean, it sees some of these cells, but overall does a quite poor job. And this is after our improvement, where it actually can automatically find the cells in this image. Right, we then compared this to the original Segment Anything model in quite some detail, also studying this interactive segmentation. So always you see here, the cyan dot is kind of the input to the model, telling it, okay, segment this cell. Then red is the output by the model, and yellow is a true cell mask. And we see left is original SAM, here's our microsam, and we see across the board for all of these imaging conditions, it improves significantly and kind of can pick out single cells from these clusters. Right, similarly, we tested it for a lot of conditions for automatic segmentation, where we just get in the image without additional input and just tell it to segment all cells or also other structures like mitochondria and EM, and also see that it's very accurate for all of these cases. So, and based on this, we've built this tool, also called microsam, right, where in a very user-friendly Nampari plugin, users can just plug in their images, analyze them with all of these methods, right? So for example, here you see this interactive segmentation with these box outlines for cells, and here you see that we can also segment interactively mitochondria in 3D. So all of this is available, already quite widely used, but of course this is still not solving everything and comes with some limitations. So the main thing here is that we have quite a naive extension to 3D. So the original model is 2D, and to extend it to 3D, we just process things slice by slice and merge the results. This works good for relatively easy shapes like mitochondria, but for more complex branching shapes, this really has its limitations. Also, the model requires a lot of resources because it's larger than traditional deep learning models. Luckily, Facebook Research, who developed these underlying models, still has not been sitting idly, and last year published SAM2, which tackles video segmentation and interactive video segmentation, right, as you see here, for segmenting this football and the foot of the player, or even here for tracking bacteria in microscopy videos. And this model now can both process multidimensional data natively and is also more efficient. And so now this brings us to our preliminary work on now adapting SAM2 to build microSAM2. So really a model that can automatically and interactively segment multidimensional microscopy data. So see how this looks as a sketch here. I don't want to go into details, but basically the idea is a model that can both work with 2D images of cells or other structures and 3D volumes and then even videos and give us accurate segmentations either automatically and interactively. And now we have a first working preliminary model for our internal purposes, trained on 3,500 images or volumes for over 600 annotated cells and nuclei. And for this conference, we just thought, well, let's see how well this does for some of the latest expansion microscopy data. So here you see this for sort of a small volume from this holistic data, right? So whole zebrafish in XEM, see the segmentation results in 2D and how this looks in 3D. So already does a pretty good job. It's far from perfect, but it's on a good way. And also, right, we can do this interactively, right? So just from a single point, segment these very, very clustered, dense nuclei in the volume. And we tried it, again, with the same model, also for the holistic data, right? You see a zoom in for this here, and then apply this model to segment kind of these neurites in there, right? So in 2D, we see that this is very complex data, but in 3D, if we just highlight some of these processes, again, in a very small volume, but we see that this does quite an accurate job in finding kind of these neural traces in there. Again, also interactive works in there. So this was a quick overview of this work. Huge thanks goes to my student Andre, who've implemented all of this. And if you want to know more, you can also go to his poster, which discusses these applications and our work in more detail. So in the last 1 minute, just a quick teaser, right? So the next frontier is then what do we do at the nanoscale, right? So what do we do to analyze proteins in expansion microscopy, right? So this shows the work already done in the paper by Ali, where what we can do now is basically take these images for a single purified protein, feed them into this autoencoder, and get out the 3D structures of single proteins or complexes. Right? One of the big goals there is to move into cellular context, right? Where instead of just having a single purified protein, we also need to distinguish proteins and complexes from each other. And just as a very quick teaser, right, we're currently developing this for cryo-electron tomography. What we do there is develop these methods that can both localize and identify proteins, and that can be trained based on synthetic data and then be adapted to real data and yielding similar results than experimentally trained models. Not time to go into any details, but this is sort of our goal here, to develop these systems that can do this both in one day, in cryo-ET, and eventually also in in situ one data. Right, so just as a quick conclusion, right, on the term of segmentation, I've showed this microsam model. Right, if you're interested in more, you can come to the poster. Also check out this link. It's already in quite wide use. Right now we are developing microsam2, which will extend this to more complex multidimensional data. And then the other big goal that we have is to move to visual proteomics by analyzing proteins both in cryo-ET and in one. Right, and lastly, thanks to all the collaboration partners here who made this work possible, my group, and then the funders of this work. And thanks for your attention.
So thank you, Constantin, for this brilliant work. We'll have one question because we're 5 minutes late and we want to have the lunch served warm, not like yesterday cold. So please go ahead.
Hi, great work, Constantin. I was wondering if you are applying to any of these, especially the last one, to subcellular organelle type of segmentation too?
Yes, right. So that's both for the, basically that's still in this first microsam work, where our goal is to extend this also to subcellular organelles. We have done some work on EM already and really want to extend this. It's a bit limited by the available training data, but we are also working, especially with the EM community, to expand this.
Okay, thank you very much, Ali, for the introduction, also for the invitation to come here. So it's really a pleasure to be at the meeting where I'm not the only one doing expansion microscopy. Still, when you go to biological meetings, you're often the only one. But here it's very nice to see many partners in crime, and also to be able to discuss a lot of issues that we might be encountering. So I hope to have more of that later today. Because it's a technical meeting, I decided to not spend too much time on the biology, but just not go in detail, but show a lot of applications for which we use expansion microscopy, and then also focus on how do we actually calibrate our expansion factors, and how do we deal with deformations that might occur when you start swelling cells. But before I can get to the application, of course, I have to introduce a little bit of biology. As already mentioned, our group really studies how cells, and in particular differentiated cells, establish their shape and intracellular organization. And this is, to a large extent, due to the cytoskeleton, which is a polymer network of microtubules and actins, but also with associated motor proteins that can move over microtubules to drive cargo transport to specific locations within the cell. And for example, in neurons, this is really important because you see here that neurons have this very beautiful shape with multiple dendrites that receive signals from other cells, and then a single axon that sends the signal to other cells. And you see a clear morphological difference, and this reflects a difference in protein composition. And so many proteins are synthesized in the cell body and then need to be differentially located to the right destination in the cell. And this is driven by motor proteins that move either to the plus end of the microtubule or the minus end of the microtubule. And so it's really important to understand microtubule organization to understand how transport works in cells. And to make things more complicated, so microtubule, it's not just one type of microtubule. It's now really an emergent theme in the microtubule field that there can be different subsets of microtubules. And in particular in neurons, some microtubules are extremely long-lived. They barely grow and shrink, whereas regularly microtubules are very dynamic, so they grow and shrink all the time. And the interplay between microtubules and microtubule-associated proteins then can mediate that the subset of microtubules becomes highly stabilized, and because they are long-lived, they accumulate post-translational modifications, and we can detect that by staining for different markers on the microtubule. And so these microtubules are diverse, and if you want to really understand transport, you have to be therefore, it's important to resolve them really one by one, because we need to see what's their identity and what's their orientation, for example. And so as you know, so they are quite small, 25 nanometers, so we need super resolution because they are often spaced such that you can barely fit a microtubule in between two microtubules. And this actually this whole idea of polarized transport to specific localization is not just true for neurons. So here we have airway cells. Also in airway cells, you have a clear apical site and a basolateral site, and also their transport processes are polarized, so either biased towards one side or the other side, and also through an interocytes, for example, as shown here. Now, this is one of the first movies that we acquired with expansion microscopy, and this really blew my mind and convinced me that even for fragile structures like microtubules, it's really a very powerful technique, because we had been using a lot of STORM, single molecule localization microscopy techniques to get high resolution images of microtubules, but we always fail to get it in 3 dimensions. And of course, this is the beauty of expansion microscopy. You also increase the actual resolution. And so now we get these very beautiful images. And here you see actually in green the dynamic microtubules marked by tyrosination, so they grow and shrink a lot if the cell is not fixed. And the acetylated microtubules, which are highly stable and, for example, enriched in axons. So we can now zoom in, for example, in the dendrites. And since the dendrites are sort of on the cover slip surface, what we do is we cut the gels, we flip the gel so that we have the highest resolution in the cross-section of the dendrite. And then you can see that we can really start to reveal individual microtubules. And so even though we cannot reveal all microtubules, we can sort of calibrate the intensity of a single microtubule. And because we label both the modifications that mark the stable and dynamic and all the total microtubule population, we can do quantitative analysis and really study how many microtubules are there of any subset. And if we just see that the stable microtubules, the ones positive for acetylation, they mark 75% of the population, and then 25% is the tyrosinated microtubules, which already was surprising to me because it shows that we can basically account for all the microtubules by just staining for these 2 modifications, whereas it might have been true that there is perhaps a 3rd subset, but we don't see it here. So this is what we do in neurons, and we still continue to do this, and there's a lot of very interesting cell biology and microtubule-associated proteins that regulate all of this. But we also started to do it in airway cells. So on the left you see an overview. So here we use M-Cling as a membrane stain, which actually worked very well for us, especially for the plasma membrane. And then we use these total protein stains or NHS stains. And here we also switched to T-Rex, the recipe which gives us like 10-fold expansion. And then if we look at the airway cell, the microtubule network, you see that they really are also very different than the textbook picture where you have a centrosome and all the microtubules nucleate from the centrosome. It's really a linear network where all the microtubules are pointing from the apical surface down to the basolateral surface. And if we now stain for the same two modifications, stable and dynamic microtubules, actually very striking, because also here we see that they have a really different organization. So the stable ones, they form actually their own network, which does not have this polarized apical to basolateral organization, but has a different organization because they have different functions and maybe mediate the planar cell polarity. So this is very interesting, but then we became, of course, interested to where do these microtubules nucleate from. And then Emma van Grinsven, she did most of the work in airway cells. She found that actually you have these basal bodies that nucleate the microtubules for the cilia that move. But if you look at the basal foot, actually very often microtubules emerge from the basal foot. So then we became interested in the molecular composition of this structure, and we started to use this stain for many, many proteins known to localize to centrioles. And we could actually, because there are so many basal bodies in these airway cells, we can just analyze many of them and sort of distill an average distribution. So here you see for 3 different proteins and the total protein stain, the distribution. We can reveal the 9-fold symmetry. And you can really see that they all have a very distinct localization. And what Emma did, she actually tested 30 different proteins that are all localized to this structure. And she could really give them, pinpoint them very at specific places. And good for us is that we found that cametubulin, which is the nucleator of microtubules, indeed was enriched also at this basal foot, so which then explains why microtubules can be nucleated there. So this was on a 2-dimensional monolayer grown on filters. So we now move more towards 3-dimensional model systems. So here's an example of a mouse intestinal organoids. So for us as cell biologists, it's very interesting because you start with stem cells and then this develops into a sort of tissue mimics where you have very interesting cell types that are very difficult to culture and are typically quite rare. But because you culture them in organoids, you can really get this very interesting differentiated cells. And so here you see an expansion microscopy image of a crypt. And you see already in the ultrastructure differences between the different cells. So and if we just cut out, we can actually very easily identify which cell type we're looking at by in the pan stain, because if you compare it with electron microscopy, it looks basically the same. So this is very interesting. But we can also confirm, of course, cellular identity by staining for proteins known to be important for the specific functions of these cells. So here we stain for stem cells, or here we stain for Paneth cells. And what we can also do is use all these modern segmentation tools to really segment every cell in the whole organoid. And then we really get the morphology of the cell, but also we can really look at the cell type specific distribution of organelles and the different microtubule organizations in different cell types within this organoid. But I will not go into detail. I will just give one more example here. So here we have the enterocytes. So these are really the cells that absorb the nutrient from your stomach because they have these microvilli, actin-based structures. But what is possible if you have a mutation so that you don't have myosin 5b, then you get microvillus atrophy. So you see that the microvilli are less abundant, are also shorter, and you get these inclusions called microvilli inclusions. So if you have this mutation, you will develop microvillus inclusion disease. And here's another example. So we can beautifully see this, and normally this is a disease that needs to be diagnosed with an electron microscope. But of course, if you now want to analyze this and want to measure the size of the microvilli to really see how severe the phenotype is, then it becomes really important that you sort of develop some trust in the quantitative measure of expansion microscopy. And I think this is something that has not been discussed a lot, is that, okay, how exactly do you know your expansion factor Because I think the common practice used to be you take a ruler and you measure a gel before and after expansion. And so when we ask 10 people in the lab to do it, you get already a 10% variation in expansion factor. But what's even more so, if you do iterative expansion, then it becomes quite complex because maybe sometimes you cut parts of the gel that you then further expand, and then you cannot use this trick anymore. So you have to do something else. Now, I've been teaching microscopy for about 15 years, and when talking about calibration, I always was joking that in the old days, people would use the size of the nucleus to map out what 10 micrometers is. And that is, of course, not a good practice. But then over the years, I realized that this became actually the standard practice in our field because there was nothing else. So to sort of not lose my credibility as a teacher, we had to develop a new technique where we could be a bit more quantitative and reliable about what is the actual expansion factor. So what we did is we developed GelMap. This was developed by Hugo Damstra and Josiah Pasmar, who are both in the audience. And it uses microlithography. So what we do is we coat cover slips with protein, and then we use a photomask and UV light to burn away the protein in most places so that we can impose all kinds of patterns on the cover slip. This is a very standard technique. You can also shape your cells with this, for example. So we had this already ongoing in the lab, but we now designed photomasks that were tailored to this idea that we can make a reference map on the cover slip. And since this is protein-based, it will undergo the exact same chemistry of the rest of your cell. So it will be incorporated in the gel and will expand with the gel. And if all goes well, then it will remain squares. So then you know that your gel is doing well. So here you see an example of a gel map, a gel with GelMap that it has been expanded, and depending on how you think about this, you can be very positive or a bit more less positive. So if you look here, it's most of the squares are still squares, so the expansion is relatively isotropic. But if you are a bit more negative, you say, okay, look, the expansion factor here is twice as big as the expansion factor there, and here there are no longer squares, so there is actually quite some deformation going on. So one idea is then, okay, maybe you should not image the edges of the gel. So let's focus on the middle of the gel. But if you look in the middle of the gel, so there you see that depending on your gel and the way you handle it, it can be more or less deformed. Now, I can imagine that in many cases you think, well, the deformation is not we are mostly looking at overall morphology, so we can deal with this. But still, it's important. What it anyway in all cases provides you is that you have now the dimension, so you know exactly what the expansion factor is. And if you at some point want to analyze your segmented data set to get like a distribution of spine sizes or whatever, then I think it's important that you have the capability to make sure that your calibration is really correct throughout your whole sample. So here's why. So this is, of course, the limitation of this whole approach is that this, so far, we have the grid on glass. So for cultured cells on glass, this works beautifully. If you have tissue, you have to do something else. So what we can do is we can print this pattern on the tissue, but still, of course, the grid will be on the edge of the tissue. So what we do here is, if you then refocus to a deeper plane, then you can superimpose the grid, but then you have to assume that the deformation in that place was the same. I think this is a relatively good assumption because most of the deformation come from shearing the gel, which is then assumed to be more homogeneous in the C position. But of course, this is something we now try to actively work on. But at least we now have a way to, within tissue, and even if we do iterative expansion microscopy, to really have a calibration embedded within the gel, and to also make sure that we can do correct for any deformations. So 2 developments that we are currently working on. First of all the corrections we did so far was using big warp, which means that you have to put a lot of landmarks and then correct for them. So this requires some manual labor. And so we're now working with image analysis specialists to really do it more automated. And so there are already many algorithms from the medical world that can be implemented to really put this just as an input and then already correct pretty well. And so this is something we are developing so that we can really provide a plugin that you can use very simply to put in the deformation, the ground truth of the grid, the deformation of the grid, and your acquired data, and so that will be corrected. So this will come at some point. And of course, this whole idea of 3-dimensional grids is interesting. So what we are currently trying is really for tissue work to really make sure that the grid is not just at the surface, but throughout the whole gel. And one idea that we have for this, so we already tried this, is if we implement a photoactivatable fluorophore in the gel, and then we can use photoactivation to make a pattern that is not just at the surface but in the whole gel. So that we have tried this already some time ago, and we were relatively effective in photoactivating patterns in the gel, but then most of it got diluted when we expanded. And here we still were dependent on like microscope-based activation. So I think the better way to go would be to have a better fluorophore or some amplification strategy, and then use a photomask also for the photoactivation, so that you can really do it in one go. Put your gel on the photomask, burn the pattern into your gel, and then start with expansion. So this is something we are developing. So these were the two things I promised to cover. I want to cover one more thing. Is that okay? We're celebrating how great expansion microscopy is, but I think not all the time your PhD student or postdoc agrees, because there can be periods of time where the gel doesn't work or the stainings don't work. So I think it's actually a very good place here to actually discuss the issues that we have, because normally if you are the only one doing expansion microscopy, you have no one to talk to at the conference. But now here we should be talking actually about the things that don't work. So in our experience, we have the problems we suffer with most is, okay, when we do a great staining and then expand, still we often lose fluorescence. Now, the way around this is to do post-expansion labeling. But then we have variable success with post-expansion labeling. So there's a set of antibodies that you can use, and it works. But we are cell biologists, so sometimes we just want to focus on protein A, B, and C, and that's where the science brings us. And all of a sudden, our stainings no longer work. So I was very happy to hear that you are testing now like thousands of antibodies. And so I think that is very, very useful to have. Well, of course, also there are so many protocols emerging, which is normal because we are a young field and things can probably be improved. But if a PhD student embarks on a certain project, on a certain cell type, at some point they need to convert on a recipe, and it might not be the ideal recipe. So it's always tempting to then shift and shift, but then you have to start overall the whole science again. So this is something we try to find a balance. And then finally, the imaging times. This is also, if you expand 10 times, then you have 1,000 times more volume. So the imaging slots take like whole days. So we had to buy already 2 new confocal microscopes to facilitate all our expansion microscopy. So this is not really good for democratizing high-resolution imaging. So, of course, it would be nice if we can expand so much that at some point we just look at it and can bring our sample to the conference to show it. But as long as we still need microscopes, I think light sheet microscopy is a way to really address this. So also nice that we saw some of that. We also tried it a bit. We used a commercial lattice light sheet from Zeiss. And what's interesting, it's a stage scanning microscope. And that is quite interesting. So because the stage can just move and move and move and move. And so normally this would be a field of view that we image. So this is imaged on the lattice light sheet. The resolution is a bit worse than on our best confocals. But now when we just scan the stage, we can image and really image like a full centimeter of gel. And this was done in 5 minutes. And this would have taken like maybe 5 days on a confocal microscope. So I think in combination with higher expansion factors, this should be actually the go-to method at some point for many of our applications. So in summary, to thank the people that did work, everything in bold is someone I showed the data from. Emma did most of the airway work, and the gel map was then developed by Yuko and Josiah. Thank you very much.
Thank you, Lukas, for this fascinating talk. We have room for one question. Donatos?
Hello, nice talk. I was wondering whether you ever expanded just the grids to know whether these distortions, especially at the center, come from the gel chemistry itself or...
Yeah, so actually the example I showed with the whole gel that was expanded, that was exactly that. Yeah. So it's not just because of the biology, it's also because of the whole gel.
Okay.
Silvio?
Another question.
So we'll have a second question. I'm sorry, I can overrule the first comment.
Question 1B.
Lukas, one great point about this is
it's fantastic to have a grid. The idea of having a grid that you can actually draw with a laser within your sample, that's even better. But I'm still stuck for a method that would check loose ends of gels and breaks on the molecular scale, or rather on the molecular scale of the expanded molecule. So why not label the gel itself completely in one of the channels and try to image essentially the gel structure in one of the channels? I mean, you know, would that be like the density is too high for any technique imaginable, or what's the problem?
Well, so of course, for us, the key reason to develop this was that we wanted to know the expansion factor. So I think there you have to have some kind of periodic pattern that then expands, and so you can tell it. Of course, another separate question is, okay, it's very exciting to understand much more about what happens on the gel level, and what actually, how does an expanded gel look like in all these different chemistries, the polymer networks. This is not something that we are currently like trying to pursue, but I think it should be pursued. And maybe, so of course there are all these old papers from the old days where people use EM to analyze this. There are other scattering methods to do it.
Yep.
But maybe also like high resolution fluorescence microscopy is perhaps the best way to get this resolution and this information.
Fluorescence microscopy is the best way, no matter what you want to do. So
yeah.
Okay, thanks a lot.
Thank you.
Yeah. Thank you, Lukas. Let's thank our speaker once more. And our next speaker is Dr. Ksena Longrin from the Laboratory of Molecular Biology at the Medical Research Council, Cambridge, United Kingdom. And Ksena will be speaking about expansion of Toxoplasma gondii cysts in vitro using enzymatically enhanced ultrastructural expansion microscopy. Let's welcome our speaker.
Awesome, let's crack it. Now, I'm Ksena and I'm a postdoc in Rodriguez lab at the Laboratory of Molecular Biology in Cambridge at the moment. And today I wanted to share with you a neat trick we developed to enable expansion on heavily glycosylated toxoplasma cysts that you see here in blue residing in the huge fibroblast cell imaged with light sheet. Now, oh, it doesn't change. It doesn't work. Yes. Yeah, I do. It worked 5 minutes ago. Should I open it again? Let's close and open it again. I think it should work now. Yes, great. Okay, cool. So, Toxoplasma gondii, the most successful parasite on Earth. Toxoplasma infects multiple cell types in all warm-blooded animals, including humans, and in fact, it infects 1/3 of human population worldwide. Now, interestingly for us, this parasite during the chronic stage can form a cyst around itself and in such a way contributing to long-term infections. And in vivo, it likes to sit in your brain, so in post-mitotic neuronal cells and skeletal muscle cells. And once converted, the cyst wall surrounding the parasite becomes the interface of host-pathogen interaction. So why should we care? Because chronic stage is not curable, and if it goes unchecked, it leads to severe complications, especially in immunocompromised people. Now, to pave the way for the drug development, we need to know which proteins are essential for the cyst wall formation and growth, and what is their subcellular location. Now, I answered the first question using site-directed mutagenesis and some high-throughput imaging, but the second one was answered using expansion microscopy, which is the focus of my today's talk. So my initial attempts at different expansion protocols didn't work on encapsulated parasites. And to give it some context, when comparing non-expanded to expanded cysts using standard ultrastructure EXM, we saw a large black gap between the cyst and the surrounding tissue of the fully expanded host cell. And in fact, when we measure the nuclear cross-section of the parasite before and after in acute and the chronic stage, we saw smaller 2.5x expansion factor in the chronic stage compared to the acute, which means regular ultrastructure expansion protocol wasn't particularly suitable for super res on the cyst wall. Now, we knew that cyst wall is heavily glycosylated and it has a tons of mucin domains. So we decided to use mucinase as the pan-selective, well, actually not pan-selective, sorry, the selective protease against the mucins. And when we added this to as an additional enzymatic digestion step to our protocol, we were able actually to fully expand cysts to the level of the acute stage and to the agreed levels of ultrastructure expansion. Now, this means that by doing this work, Toxoplasma field now have a reliable expansion super resolution technique to study chronic stage of toxoplasmosis, and affordability is a big thing in parasitology. Now, in case you wondered how the level of details look in 3D, this is how. So this is before expansion, after expansion, and the disclaimer is just two different cysts, one in fibroblasts and another one in neurons. Of course, you can do it on the same. Now, the first interesting biology that we targeted is the location of dense granule protein 2 in the relation to the cyst wall protein CST1, which I already introduced. Now, GRAD2 is a fun one, is a key to organizing tubules inside the parasite vacuole in the acute stage. And also required for organization of the cyst matrix, so inner side of the cyst, in a chronic stage. And its relocation to the cyst wall during the stage conversion suggests it's involved in shaping the structures during the conversion. Now, standard confocal images show full overlap of GRAD2 here in green and the cyst wall marker in CST1, both appearing at the smooth signal across the cyst wall. This is the cyst wall. Now, when we add mucinase to the protocol, we can actually see GRAD2 as puncta, distributed across still smooth cyst wall marker. Now, if you want to know why it's relevant and why it's cool, please talk to me to my poster. We published this with some additional quantifications and observations last year. But since then, well, this work was done at the University of Edinburgh at Joanna Young's lab, where I spent lovely 3 years unpicking parasite biology. But 4 months ago, I moved to Cambridge and started the postdoc there at the Laboratory of Molecular Biology to build imaging pipelines that reveal direct communication between neurons and immune cells. So it's an extremely interesting project. It will involve lots of whole organ organoid clearing, also a bunch of expansion microscopy techniques on which I already started, but unfortunately it's quite short. So if you know anyone who would benefit from this kind of expertise, I'm open for postdocs, especially in Switzerland, and I will be available from next May. Thank you.
Thank you, Ksena, for this wonderful talk. One question? We have one?
Hi, thank you for the nice talk. This is, I guess, more of like a biological question because I was just curious, you mentioned at the beginning 1/3 of the population is infected with it, but I guess not everybody is obviously at the chronic stage of it. So I assume so. This is a very successful a parasite. Have you thought about actually putting drugs inside the parasite that can be delivered, because it seems like it can cross into the brain, right? You said it's
yeah.
I don't know
actually,
anything about
it's a cool thing that you've pinpointed, because toxoplasma at the moment is in development as the drug delivery agent.
Okay.
But it's really hard to do because attenuating this thing is a hard stuff. So we have an attenuated strain with the reduced virulence, but at the moment that we couldn't move further to the clinical studies because we need to figure out which parts of the organism are affected by the parasite.
Thank you.
Thanks.
Everything is getting set up. So Professor Nadia A. Smith is from here in University of Göttingen, and she works and her research focuses on smart drugs, biochemical probes, and labels. And that is what she's going to tell us about with her talk titled Tuning Labels, Probes, Labels, Linkers, Anchor, and we can now proceed to the talk.
Almost. No, I don't, but I could get it if needed. Okay. That looks good. Yes, thank you so much. And thanks a lot for the very kind introduction. Thanks to Silvio and Ali for inviting me to speak here. It has been an absolute blast so far. I'm feeling a bit intimidated because I'm going to speak to you as you see it down there, as a chemist. So we are a group in the Department of Chemistry here in Göttingen campus. We're going to turn 4 years old next Wednesday, so I would still consider us a quite young lab. And this is built of a bunch of fantastic people, which are quite interdisciplinary. So we have synthetic organic chemists, we have photochemists in the group, we have people who consider themselves a bit more of biochemists in background, and we try to use this diversity to blend together our research program, which is split on different pillars, but all going down to actually making chromophores and using these chromophores to answer different questions in biology. So what we are interested in is to make so-called responsive molecules, so molecules that if you shine light or different stimulus that change their properties so that they can give you a response at a system level. We like to incorporate them into larger structures such as lipids or proteins to make those move upon adding such a stimulus. We like to use different colors for that, so we work on something that's called chromoselective photochemistry, and more I think interesting for this community is our research program on smart labeling and click chemistry, which I'm going to talk about today as well. Now, to give you a bit of a flavor, what the lab is doing, the first part is not on labeling chemistry, but on actually using light to control pharmaceuticals, bioactivity, and stimuli in general. So if you ask AI what this research is about, you get this picture of a cage with a drug inside, and then I ask it to be light activated. So this is the broader picture. Let me explain you what that means for us. So when we think about light activation of bioactive molecules, we think very simply about a molecule that can temporarily block the activity, so render our probe, our drug inactive, and upon irradiation, you would change the structure a tiny bit, and you would make the compound active, if you will. Now, a simple example. I brought you here ephedrine. This is the black structure, and you see here in blue a so-called light-sensitive protecting group, our cage. And the only thing you have to understand from that structure is that they are roughly equal in size, so you can imagine if you want to address a biomacromolecule, so recruit a receptor or an enzyme, this won't work anymore if your molecule doubles in size. So upon irradiation, you now can cleave off this photolabel stopper. You have a bit of waste product that can be washed out. And in this case, you liberate the bioactive compound, which is ephedrine. And ephedrine then can be measured, so you can measure the response on the system, and we did it together with the group of Toby Brückmann here at the UMG in Göttingen, and you can basically influence the contraction rate of cardiomyocyte on a plate, and we're always moving that towards the tissue and small organ level. You can take the very same type of chemistry, but attach now these long lipophilic chains which force your compound to be incorporated into a lipid membrane. So now you can anchor and position this molecule, and you can still address it with light. In this case, we attached a positive charge on the original photolipid so that we have overall a positively charged membrane surface. We measured that to be 42 millivolt, and upon stepwise irradiation with light, we could cleave off this positive charge. We change the potential of the membrane surface, and in the end we reached a potential of minus 14 millivolt. So we inverted the surface potential of our membranes, and we are applying that to fusion processes. Now, why this was all about activation, we of course also want to be a bit more flexible and turn the whole thing around. So we also work on deactivation, where we first have a molecule that is generating some response, and then we want to be able to switch that off. And for that, we looked into the family of capsaicinoids. This is nonivamide, an example of that. And we simply chemically attached a nitro group, which makes this whole molecule susceptible to light, so that before irradiation, if you take this molecule, you get a response on isolated neurons. This is a patch clamp experiment where we recorded the voltage over 2 different neurons, and also a docking experiment showed that indeed we bind to the same receptor. Then if you irradiate and you break this molecule, you basically generate 2 equally large fragments, and you don't get any response anymore. So again, we can use the same type of chemistry to get a whole diversity of different tools for biochemical and biological research. Now, we don't want to stop here. I would like to share briefly where we really want to go in the future, and this is to build up our dream toolbox, which is consisting out of not only one stimuli targeting one different function, but a whole range of different biologically relevant stimuli. I just listed a few of them. And I'm not only using one color, such as shown on the slide before, but also combining that with different colors. So in the end, we can come from the outside, just irradiate with different colors, and generate a whole diverse response on a cellular level. Now, if you think about that, the only thing what we would need to be able to do is to split our UV-Vis absorption spectrum into different ranges, and assign, for instance, one range to one reaction and a second range, in this example, the blue range, to a second reaction. And from an optical point of view, so if you want to buy LEDs or lasers, you can just do that. So this is not particularly challenging, but a challenge lies in the design of chromophores. As you can see here, in an absorption spectrum of different chromophores we made in the lab, in the visible light range, you have quite isolated bands, so it's very easy to buy an LED that matches this band, and then you can address this molecule selectively. But here in the blue and UV range, they start to overlap, so trying to generate selective recruitment of one of these chromophores without hitting a second one is really not easy. So we set out to try to solve this problem. And we wanted to do that first at what we thought at the time, a very simple example, namely the dosing of protons, or in other words, the change of pH through remote control. And for that, we made a few molecules. On the top, you see molecules that liberate acid. You see all acetic acid molecules up here, and the bottom ones liberate base, piperidine and DBU, for the ones who are interested in that. So we took these molecules, we tested first if they are actually generating the function, and indeed, if you look at 2 of them, and you see in orange the light pulses that we exerted on the system, you see that either the pH is going down stepwise or it's going up stepwise. And I hope you can appreciate that this is a very predictable reaction, because shorter irradiation times give you a smaller change than longer ones. So just from changing the irradiation intensity and duration, you can really adjust now the pH from the outside of the system. Now, the interesting part of the experiment came in the next step, because we, of course, didn't want to just have a one-side response. We wanted to mix that and see if we can get an overall control. So we mixed a base donor and an acid donor in a 1-to-1 ratio together, so that with one wavelength we would generate base, and with a second wavelength, in this case blue light, you would generate acid. And indeed, when then irradiating and toggling with two different colors of light, depending on the color which we were shining, we were either increasing or decreasing the pH. And again, that was in a dose-specific manner, so longer times gave us bigger changes than shorter times. So we wrapped up that story, and just wanted to give you a glimpse on that. And now I think I'm going to hopefully touch the topic which is more relevant to the field, and this is our work on optical imaging with smart probes. And again, I brought you a picture what DPI thinks that could be. Okay. We are working in the context of fluorogenic optical probes. So just to bring you a comparison, this is what many of you already showed. This is classical labeling with fluorescent probes. So two mainstream examples is a coumarin-based dye and a body P-based dye. A lot of them are commercially available in different ways, and many of them are functionalized with NHS ester as a labeling group, and we saw several examples where those had been used. Now, these are permanent fluorophores, so they will be fluorescent the moment you have them in the bottle. The difference to fluorogenic probes is the fluorogenic probes are only becoming fluorescent upon response to environmental stimulus. There can be a fluorogen introducing a structural change, there can be an unquenching reaction, polarity change, or a conformational lock, which we often see in fluorophores that are specific for membranes but not for the cytosol. So this is the overall principle, and the nice thing is you will have an answer into your system. So you don't only have a fluorescent system, but you have a responsive system. We became interested in that when my PhD student, Dong Chen, now I think almost 3 years ago, found a reaction between a pyridine aldehyde over here with primary amines that gave us a weak fluorescent signal. Now, I want to spare you the mechanism. I just want to highlight that comes from the Journal of Educational Chemistry, so quite broadly applicable. And you can react that with lysine, in the end forming this compound here, having a permanent positive charge. We next wanted to see how we can influence the fluorescent properties. And on the one hand, you can do a pi extension, so you just make the pi system larger. That gives you a bathochromic shift of the system, so already absorbing and emitting a bit more in the visible range. And the second thing we did, as this had a positive charge, we attached push substituents on different sides of the aromatic core, again, tuning spectral features. Just want to very briefly show you that those compounds are not straightforward to make. This is the synthesis example of one core to assemble only one of these derivatives with the pyridine aldehyde warhead on one side and the push substituent on the other side. And of course, we wanted to diversify that, so we didn't only do that with one push substituent, but with different ones. And that was really not easy because we got a lot of side reactivity attaching our push substituent on the wrong position if we didn't do the synthesis carefully. We also modulated the different positions, took two different cores, again with different positions, to in the end derived the final molecules that could react with a primary amine to form our fluorophore in situ. We studied the overall range, and I just give you the results of the overall study. So in one position we found this gave us the largest Stokes shift overall. The position on the other side here gave us the overall highest quantum yields, which is important for the brightness of the fluorophore, but this turned out to be synthetically the most challenging to make, so we in the end settled on middle ground with a medium-high quantum yield and then optimized it further by tuning the ring size. It then in the end now gave us a fluorophore that had quite high contrast, so it was brightening up a lot upon labeling. The soak shift is around 12,000 reciprocal centimeters, so you irradiate you excite in the UV range and you record in the orange range. We have a quantum yield of up to 30%, and this gives us an overall brightness which is really compatible with microscopy. We could label diverse functional groups, so peptides, sugars, different lipids, and also halo tags. And we then went on for imaging, just bring you a preliminary result. This is protein labeling directly. We used a nanobody in collaboration with the Opazzo lab. This is now imaging with the alpha conjugate down here at nanobodies immobilized on beads. And now with our very new compound, which has solubilizing groups, we are currently labeling larger proteins and perform fixed and live cell experiments to wrap up the whole story. with that, I would like to wrap up today's talk. Oops. So we are generally interested in making dyes and especially fluorophores smarter, and this could not be done without the help of my great group, the support of our funding agencies. I want to highlight here the SFB 1286, which has been incremental for the labeling project, and I of course would like to thank all of you for listening and your kind attention.
Thank you. We can take one quick question. No? Then let's thank. Oh, okay.
So one obvious question, so are you going to try an expansion microscopy? Do you think that this could be our new NHS-STED fluorophore?
Yes, I actually want to say that, but I got super nervous when my chair got up, so I forgot that. This would be the next obvious step. So we would really be interested in seeing how, so it's a dye which is selective for the same amino acids as NHS. So it's having the back-to-back comparison, especially in the context of expansion microscopy, would be interesting to see if we get a different labeling density. And this is something which we are talking about with Ali and Silvio. Thanks a lot. Yeah, this was the last part I wanted to say.
I'm sorry. But thank you so much for your talk.
Thanks.
And we can move on to the next.
The University Medical Center. He will talk about seeing the unseen and a bit of pathology, right, Tiago?
Here? Yeah.
Thank you very much for being here. The
Thank
stage is
you.
yours. Thank you so much.
I want to, of course, start by thanking Silvio and Ali for the invitation to speak to all of you at this fantastic meeting. I've been learning a lot. You will see I'm not a microscopy specialist. I'm a user. And the way we've been using is to try to understand a problem that is as old as life itself. And this has to do with understanding how different factors affect the folding, the phase separation, and misfolding of proteins. This is very, very relevant in biology and in disease. And this, in the particular case of neurodegenerative diseases like Parkinson's disease, this is highly relevant, and I'll try to explain to you why. So, so Parkinson's disease is a complex disease characterized by well-defined clinical symptoms, but it's very complex with multiple alterations that we still don't understand in detail. What we think is happening is that there is the loss of dopaminergic neurons from a region in the brain called substantia nigra, and the typical neuropathological hallmark is the accumulation of protein aggregates that are called Lewy bodies. And as a basic scientist working in this field, it has been a bit frustrating to think that although James Parkinson's described the disease more than 200 years ago, we still cannot treat it very well. And I've been thinking about this, why can't we treat it better than we've been doing so far? And of course, there's many possible explanations. The need for additional funding to translate the findings from basic science into clinical applications is a limitation. But I would like to convince you that this is also because we do not fully understand the underlying molecular mechanisms. And one important mechanism has to do with this process that we tend to represent in a linear manner. This is an oversimplification, but this is how we understand this process of starting with a protein that in this case, in the case of Parkinson's, is called alpha-synuclein. And this protein at some point starts to misfold and to accumulate and form higher order structures that eventually, in the case of Parkinson's, are called Lewy bodies. What the problem is that we don't know which of these species we should target to treat Parkinson's disease and diseases where there's the accumulation of proteins. And this is more complex than what was represented in the previous schematic, but the way we think of it is that there's a biochemical phase of the disease where proteins start to assemble and to form the inclusions. At some point, the accumulation of these inclusions inside the cell leads to cellular alterations, and I call this phase here the cellular phase of Parkinson's disease, and eventually, once different circuits get affected, the clinical manifestations become more severe, and then it's really difficult to treat the disease. So we are very interested in studying the cellular phase of Parkinson's disease. And since 1912, when Friedrich Levy used his very fancy microscope at the time, that we've known about the different structures that alpha-synuclein assemblies can take in the brains of patients with Parkinson's. Now, when a neuropathologist looks at the brain of someone with Parkinson's, normally they see the Lewy bodies. So these are easy to see. These are very large. They pose no challenge. But we know there are some cases of Parkinson's, some genetic cases, where when they look, they don't really see those alpha-synuclein inclusions. And this has been puzzling. We don't know how is it possible that you have disease without having those typical protein aggregates. And we want to understand this in more detail. So the question is, can we detect different types of assemblies beyond just the large species that have been seen since more than 100 years? And can we detect these smaller species that we think might be even more toxic and more related to disease? And recently there were 2 publications in Acta Neuropathologica where they used proximity ligation assay to show us that in the brains of a patient that carries the LRRK2 mutation, one of the genetic forms, that does not have Lewy bodies, but using PLA, they could detect alpha-synuclein assemblies that no one knew were there before. So we've been trying to model this process in vitro using simple cell models and using bimolecular fluorescence complementation, fusing alpha-synuclein and alpha-synuclein related proteins and interacting proteins. We've been able to develop a model whereby we can see these inclusions in living cells. So again, this is easy to see with a conventional epifluorescence microscope. We showed that we can see this also in primary neurons. We've used FRAP to characterize the dynamics of these inclusions to show that some of the inclusions that we can identify, they phase separate, whereas others are more immobile and don't recover so rapidly. We've done biochemical characterization. I'll go very fast over this. But what I want to show you is that now, using super resolution microscopy, we can see different types of these species. And here we looked at different mutants of alpha-synuclein, so we can see some large objects, no big deal, we don't need STED for that. But what we need super resolution for is for some types of species that now we start to see and that we had no idea were there. And using expansion, we started to use expansion already a couple of years ago with the help of Silvio. And we showed that we could expand these cells and we can see these inclusions. And similarly to what we could see with STED, now we can see species that we didn't know were there. And we can ask whether some of these are more toxic than others. So this is the big question we want to address. We've been doing this also in human brain tissue. We've been expanding human brain tissue. And this is just a simple example to show you that we can expand a Lewy body into something much larger. And now, using different markers, we've been looking at the different types of assemblies in relation to other cellular markers to try to understand the biology behind the formation of these inclusions. So what lies ahead is that now we think that using our understanding of these different assemblies of alpha-synuclein, we might be able to classify Parkinson's disease patients better than what we've been doing before. And Silvio and Ali showed that they can detect some of the oligomeric species in CSF and maybe even in blood samples of patients. And now we have some new nanobodies that we are using to try to study both human samples and also the cellular models. I don't have images to show you today, but this is what we are working on at the moment with new nanobodies that we hope will enable us to see these different species of alpha-synuclein. So to summarize, just 2 simple takeaway messages. One is that we are modeling the cellular phase of Parkinson's disease to understand the underlying mechanisms behind Parkinson's, and we think that using super resolution microscopy will enable us to identify species that will be more relevant in the context of disease. So I'll stop here. I'll thank the people in my lab that contributed to this work. Tamer, who is around, is doing a lot of this work using the nanobodies, so you can talk to him about this, and I thank you for your attention.
Thank you very much. We have time for questions.
Yeah, so very naive question. I'm not part of this field, but I remember one of the papers showing a claim study of the Lewy bodies where it showed in electron microscopy that there is these bodies where there's no alpha-synuclein fibers inside, like it's more like mitochondria destroyed. Do you see something similar? Or?
Yeah, that's right. So there was a paper using STED microscopy where they used different antibodies for different types of Lewy bodies precisely because depending on the brain region where you look, they may have different compositions. And indeed, they showed that they could find mitochondria and membranes and lysosomal components within certain types of Lewy bodies. So in our work, we see similar things in cell models and also in the human brain tissue using expansion. We can see different components in some types of Lewy bodies. Yep.
Maybe I have a very quick question for you, Tiago. We have seen that expansion can actually reveal epitopes that otherwise are not seen. Have you seen this happening in your cells when you have aggregates?
Yeah, we think we have seen that because otherwise these inclusions tend to be very dense, and it's true that traditional antibodies may not reach the inner components of the Lewy body. So we are seeing now in some inclusions that I couldn't show you today that they are composed by vimentin and different types of neurofilament proteins that we had no idea were influencing the biology of these inclusions. So we are seeing that.
Perfect. Thank you so much. We are doing good on time. So let's thank again our speaker. While we set up, so the next speaker is Hans Blom. Hans did his PhD in Uppsala. He also had a bit of experience in Bonn and I think also in Yokohama for a while. And now he's head of the Integrated Microscopy Unit at the Royal Institute of Technology. He will talk about expansion microscopy support and applications. Thank you very much for being here.
Okay. Yes. Okay. Yeah, thank you. So I will put on a hat that has not been presented here before. I'm running an imaging facility in Stockholm, so I'm supposed to then, to the users, support them with all the methods that you now have developed. So that's on my shoulders to kind of do. And as you see here, I'm a brother in arms with Silvio. We picked the same pictures. Over here you see the staff that is doing this job in Stockholm. So we belong to the Royal Institute of Technology, and then there's a national institute called Science for Life Laboratories. So everybody that are in need of advanced microscopy methods or sequencing methods or mass spec methods, they can go to this institute and then they get that support. And here you see a typical user getting some support. That's the Swedish king, by the way. So. Yes, so let's see here then. So we have kind of focused on a couple of areas where we give the support. My background is in single molecule localization, microscopy, STED, FCS, and all those things. So of course, when I was asked to set up this lab, I said, okay, let's go for super resolution microscopy support. Then we recruited very smart people helping out with volumetric imaging, clearing, expansion, fast volumetric imaging, building microscopes, tracking single molecules, figuring out how they are, how many there are, and things like that. And then, of course, nanoscale visualization. So, and my team then helps me and the user with this. And of course we are involved in several different networks to do this, on a Swedish network, on a Nordic network, and then of course this pan-European network. So anybody that wants support with anything that we can support with, you just contact us through the whatever network you can reach us, and then we will support you with that and train you, for example, in expansion microscopy. And as Silvio said yesterday, I actually organized a few of these expansion events through time here. So the first one I ran a couple of years before the pandemic, and then a couple afters. And I had Ed talking, I had Sven talking, I have Helge talking, I had Hugo talking, Ons talking, Virgin talking, and a lot of different people here. And 2 years ago, when Ali was in Stockholm, I asked him, do you want to take over this? Because I wanted to see a new place. So that's why we ended up in Göttingen. So yes. And there was, let's see if I can go back. I tried to do this because the users need to hear about the technology. They need to learn about the technology. Then, of course, we have hands-on lecture, usually in connection to these events, where we bring in also experts that can train students or postdoc or users. So it's been a really nice possibility to also build networks and things like that. But now we're in Göttingen, and hopefully we can continue being here in Göttingen for some years then. So, but we kind of focus on 3 areas that is, of course, of relevance: cell expansion, spheroid expansion, and tissue expansion. And my co-worker Anna, she is, so to speak, behind the cell expansion support. My colleague Steve, he is doing spheroid expansion, also taking care of a lot of tissue clearing and things like that. And my former student David, who is now a postdoc in the group, he has been developing a lot of tissue expansion methods and knowledge, and in this case he applied it to the kidney. So Silvia, you should change organ to look at. According to this conference, the kidney is much more important than the brain. So, yes, yeah. So, and we usually kind of try simple, basic protocol to transfer knowledge to users. So this week we actually had a researcher visiting from Bilbao in Spain, and Anna then runs kind of a classical 2-day-ish course where you stretch cells, and then you can then see your 5 micrometer long mitochondria or a really big nucleus or something like that. So this works really well. Yes, so here Anna is actually teaching a researcher from UK. So see how happy you are the first time you see your stretch cells. Then of course comes other problems. If it's not many cells in that gel, then of course it takes a while to find the sample. So, but typical things one kind of look at: mitochondria, nucleus, or some other organelles that you can stretch. So yes, and here are just also typical example. What can then be shown in a stretch cell that the user sees for the first time in the mitochondria. And as we are in Göttingen, you have of course done this before with STED, but now you can just do it with expansion microscopy. Or I don't know what your favorite cell is, a sperm cell. You look into that and you can see some structures of interest. And here are stretching of nucleus. So I think this is a very nice method going to help a lot of biology. It's of course kind of cool to see that the nucleus is really, really big, and you can follow this over the cell cycles, and you can figure out replication or whatever. So this was actually a group we also supported in Stockholm that just published this work. And then you can of course combine this with whatever super resolution optical method that you have, because this is chemical microscopy, so to speak, stretching samples. And then here you have an image of Steve, and he is our, so to speak, spheroid expert, and he's been looking a lot of spheroids, clearing, and also expansion, and figure out what protocols you should use and tweak and things like that. And it's been said here many times, the background goes down really a lot, so you have a much higher contrast, and then of course the resolution goes up. So when you start looking into structures, you can really resolve things, in this case in a light sheet microscope, but with confocal resolution then. And also of course, has been mentioned here, a sphere of water, it's easier than to penetrate with labeling and things like that. So that's a huge benefit. So because resolution is not maybe the most important parameter, contrast and also isotropic labeling. So. And yeah, here are just projects that Steve has supported, different things, organoids, retinal organoids or something like that. And usually you get really nice images as the contrast is really good. And yeah, the resolution helps you a bit figuring out things. And yeah, so this is David's part. So we've been doing a lot of kidney clearing. Then when Ed published his paper, David thought it was a really cool idea. So then he started stretching kidney tissue and developing a nice pipeline, trying to also optimize and making things simple. Because it was also a little bit discussed here, we have to think about time. The user cannot book 3 weeks on the microscope. It will cost them a lot, and also, of course, reagents and things like that. So some of the development should maybe be looked into, making things a little bit simpler or faster or something like that. And you don't need to maybe stretch 200 or 1,000 times. So. And here you see an image, also what Joshua showed about the glomerulus and these foot processes, and where you normally kind of look with a section in EM. And this has also been a couple of collaboration with some of the STED microscopy companies. So it doesn't matter if you use a Leica microscope or an Imperial microscope with STED, you're going to see very nice structures into these samples. And then, of course, you can do the 3D imaging. And actually, this image here we did in 2018, and it's not even highlighted in the publication from 2018 because we put it in an appendix, because we was not sure about here that these proteins that are sitting at the edge of the filtration slits in the membranes, the green one and the purple one that are on kind of one side or the other side or a little bit sticking out there, if that was an artifact of labeling or stretching or whatever. Because in the EM field it was like it should be kind of stretched over the whole slit here, which is about 50 nanometer wide. So then the question here was, why do we always see the purple molecules sitting on one side and not stretching over the full slit? And actually, just 2 years ago, there was a cryo-ET image showing that, no, there should be like a fishnet where also one of these molecules are actually just on one side. So they should be only purple on the sides, and then there should be a space in between. So with that information, one can then conclude that that nanoscale image here, which is done both by swelling the sample and using STED, so you have like a 20, 25 times higher resolution than normal. So. And this is the concluding slide then. So when the user comes, they have usually read up on some protocols, or we need to tweak the protocol for them, or tell them that, yeah, maybe we should go back to a more stable protocol or a simpler protocol. So they usually need help with that. And then if they have tried this stretching, usually they cannot find their sample. But then, of course, we tell them, yeah, we maybe need to seed more cells, or you need to have a better marker, or things like that. And sometimes it's also just, you get lost in space because you have blown up this balloon so much you don't know in what orientation you should look. Of course, it's been discussed here also, imaging distance, sample drift, but with the correct optics and maybe stable mounting, you can do this pretty good. Then, of course, imaging time and more data. Well, be patient, have a good IT department, and yeah, continue imaging. So, thanks.
Thank you very much. We are doing great on time. Questions? Maybe I have a quick curiosity, since you are there since almost the beginning of it. How much new users start to implement expansion, and is there a trend that is changing?
Yeah, it's definitely growing. We also transfer it to groups that then, when they have the knowledge, they can do it for themselves and start stretching things. And it's similar to the trend in super resolution optical, so to speak, single molecule localization microscopy. It's also the trend when they have been trained, they kind of also come back and get the support, but also it's spreading. It's not overtaking the whole imaging community, but it's growing.
Very good. Just maybe then another quick curiosity, just you were commenting on the background, right? So expansion, as we have seen, does increase signal to noise. Can you elaborate a bit more on that? What is your idea?
Well, if we just take David's examples here for the kidney, the contrast ratio here was probably, when it was not clear or expanded, then it was very hard to see something. So I would guess maybe we quantified it to be like a factor of close to 100 or something like that in contrast. So it's huge. So.
Thank you very much. I think we can proceed with our next speaker and thank the speaker. So next speaker is Jan Husken. He did his PhD at the MBL, postdoc at UCSF. He moved to Dresden at the MPI, and he's now a professor at the University Medical Center. He will tell us about high-resolution imaging across scales with modular and portable light sheet microscopy. The stage is yours. Thank you very much.
Thank you. Yeah, thanks for inviting me. It wasn't too far to come here. So in the next 10 minutes or so, I'll tell you about two things. One is the Flamingo framework that we've developed in the lab, with which we exclusively use to develop new microscopes now. And in the second part, I talk about the clear tissue implementation of the Flamingo. So the technology of choice that we use in the lab is light sheet microscopy. I don't need to introduce this here. The modern form of light sheet microscopy was something I developed back during my PhD at EMBL about 20 years ago, and we've been using this since then. And light sheet microscopy has a lot of advantages, particularly for in vivo imaging, but also for clear tissue imaging. And so just to highlight the two main features, one is high speed. We can image moving objects like a zebrafish heart at 400 frames per second in multiple colors across time and space. And we can also image developing organisms for a few hours or a few days with light sheet microscopy, something that was simply unavailable until light sheet microscopy came around. What's important to note is that all these different applications in the lab have really required different instruments. So we ended up having 10 different light sheet microscopes back at my lab at the Max Planck Institute in Dresden. And we really see ourselves nowadays as someone who built microscopes for special purposes. So half of the lab are biologists, and they basically tell us what they want, what their new vision is, and then we try to build the microscope around the sample. The problem, however, is that you end up with a lot of instruments built on big optical tables, and these have a lot of issues, namely that the most prominent one, basically when the postdoc leaves the lab, nobody knows how to use the instrument anymore. But there's many more. In particular, it's not very sustainable. It's a waste of funding to always build an instrument from scratch and basically building it in a way that it's not reproducible at all. So when I moved to the University of Wisconsin in Madison, I said this has to change. And I said, I want a reproducible microscope, an instrument that we can build many times if needed. And so the vision was to develop a modular microscopy framework that would allow us to build many microscopes. Because, as we've learned, imaging with light sheet microscopy can take hours or days. And so it would be fortunate to have many such microscopes, so you can run many experiments in parallel, and basically everybody in the lab can have their own microscopes. And so in fact, we now have 10 such flamingos in the lab, and essentially everybody has their own instrument, and we can do experiments in parallel. The other advantage would be if we can actually not just build the same, but many different variants of light sheet microscopy. As I said, this is essential because nowadays every instrument is optimized for an experiment, and you don't just have one microscope that does everything. And that, of course, would allow us to now explore a lot of new things beyond zebrafish, beyond flies, beyond whatever people traditionally use, beyond all the model organisms. And that's where things get interesting. So this is what's now known as the Flamingo. So this was a multidisciplinary design process, mainly led by these three people. And so now we have a modular microscope framework that allows us to build basically the major light sheet configurations with 2 lenses, 3 lenses, 4 lenses by just attaching illumination detection arms to a central part of the instrument around a 3D printed chamber using 3D printed sample holders, etc. So really keeping the system very flexible for all sorts of purposes. You can also reorient the entire system in space. So if this is not what you like to do with your sample, mainly having the sample hanging into a water-filled chamber, you can just take the system, turn it by 90 degrees and put your sample in a dish. So, you know, these are essentially 2 different, very expensive commercial variants, but we can switch from one to the other in half an hour. And of course, there's a gazillion of different configurations now that we specifically tailored for different applications within the lab and outside the lab. But really the most interesting that I haven't mentioned yet is that the system turns out to be portable. So by condensing everything around this unified framework, we have basically accomplished a system that we can carry with us. And so we can take this to places like Woods Hole, where we join some of the summer courses there, and we can bring the instruments to collaborators. So we're not forcing the biologists to bring their precious samples into the lab to Göttingen, but we can take the microscope wherever the biology happens. So you can do field work in the south of France, on the beach, or on an island or a boat, essentially, if you want. And this is very much in the along this idea that we proposed a few years ago, which is putting custom-built microscopes into the hands of biologists, allowing the system to iteratively switch back and forth between the engineers and the users to actually develop the perfect system, something that the biologists really need. So it's not just a system that's great for optics, but it's a system that really makes an impact in biology, and that's actually what we all want, I hope, in the end. So over the years, we've collected a ton of data, and really what I find most fascinating is this idea to leave your microscope room, leave the dark basement, and go out into the field and collaborate with people and explore new organisms that maybe have not been imaged in such detail, because these organisms and the scientists studying them might be far away from an imaging facility. And they may not have the skills to just simply build their own open source light sheet microscope or so. So the impact can be huge for us. It allows us to go to workshops, conferences, and into the lecture hall, taking these instruments, and we can do field work wherever we want. We are not constrained to Göttingen anymore, and that really allows us to also do biology not just out of an incubator, but really in the wild. So to summarize this part, Flamingo really offers tremendous benefits, not just for us, because we can very quickly now build different configurations and also follow the trend and implement what other people have done in the light sheet community, but we can also democratize access to advanced microscopy by bringing these instruments to collaborators. And I think it's a particularly nice sustainable model, especially in these times of scarce funding, where we can remodel the system very quickly and adapt it to new needs. So while this has all been mostly for live specimens, because that's where it's most important to be able to have a portable system, this system can also be built for clear tissue imaging. And Mustafa in the lab has done a tremendous work in adapting one Flamingo configuration to what we think like the best clear tissue light sheet for us, for our applications. And namely, there's been a lot of issues with the current ASLM and light sheet implementations, namely that there has not been really a system that delivers high-speed isotropic resolution. Most systems have been very slow. They had aberrated light sheets and the resolution was very limited. And so Mostafa managed to build a small compact system that allows us to use the full Flamingo framework and do ASLM. So just to remind you, in order to achieve isotropic resolution, you need to match illumination detection NA and scan the light sheet with the rolling shutter of the camera. In this way, this is not a simulation, this is real data. And there's a few special features of the system that I quickly want to go through. Namely, as you can see here, we use an air illumination lens, which also means we use an air lens in the remote focusing unit, making the system a lot easier to use. But we also have a meniscus lens on the chamber to eliminate some of the typical aberrations. The mirror in the remote focusing arm is concave, giving us a flat field, which is essential for doing ASLM and stitching. And we have a feedback system that monitors the position of the mirror, and that really gives us then ultimately this isotropic resolution. So to conclude, I just want to show you some examples, two examples. One is in the mouse brain. You can appreciate that this is true isotropic 800 nanometer resolution. And in particular, for analyzing the vasculature, it's crucial to have isotropic resolution. And here is an example of a mouse cochlea where we can trace these neurons also only because we have this isotropic resolution. So a number of cool features, and we have again 850 nanometer resolution across a 800 micron field of view at 100 frames per second. So this is 10 times faster than what people have accomplished so far. If you have any questions, technical questions, Mostafa is here in the audience. Details are on bioRxiv and very soon in Nature Biotechnology. And with this, I'd like to acknowledge people in the lab and funding, and thank you for your attention.
Thank you very much. While we switch the speaker, we have time for one quick question.
Did you bring a microscope?
No, but you're welcome to visit the lab where you can see a few flamingos there if you're interested. Yes.
This is fantastic work. Thank you so much for sharing. I have a quick question. You mentioned that you take the devices yourself to field. How easy is it for you to ship these to other labs where they can be used in resource-limited settings?
Yeah, so the flamingo goes into a pelican case. And this is something you can easily ship with FedEx, UPS, whatever. We can also remotely log into those machines via regular laptop interface, and so we can also assist people remotely in the process of setting up the instrument and calibrating it. But it's very robust, and the alignment process can really be done by anybody very quickly. So.
Great. Let's thank again Jan, and let's move on to the next speaker. So our next speaker is Lukas Schütz. He's a Chief Operation Officer at Ariadne. So Lukas studied biosciences and obtained his PhD in developmental biology at Heidelberg University. He's starting his bachelor more working on microscopy and image analysis, and then he did his PhD in the lab of Stefan Lemke, and today he's going to talk about every voxel counts, reliable training data at scale. Thank you so much for being here.
Hello, thank you very much for having me. So I guess everyone here in this room is aware of garbage in, garbage out, what that means in machine learning. And I'm very sure you spend all the effort you can on your developing your microscopes, prepping your samples, and coding your analysis pipelines. But one thing that is often neglected, and the reason is it takes time, is the training data. And if I want you to remember one number from this talk, it's 15.9. Exactly 15.9 years is the amount of time that just went into the training data of our, let's say, most important networks. I already dropped a lot of projects out when I made this number. So before I kind of go more into this number and how it originated, I want to tell you more about Ariadne. So who are we? If I talk to industry, I would say we are CRO. If I talk to academia, I could also say we are kind of an independent commercial core facility. I was 2.5 years at the ALMF at Amble, and my work today is sometimes quite similar, I have to say. So 50% of our staff are PhDs. We exist since 2014. The last 4 years we started really scaling the company, and what we do is we help you at every step of science. That can be, we can help with segmentation, so we have segmentation algorithms for all kind of light microscopy data. We make ground truth, and we also help with proofreading. So a big team of us is actually proofreading data. And if you work with us, you're in quite good company. So 25% of our clients' projects actually go to high impact journals. We have customers from all over the world, really. A lot are in the US, like the Allen Institute, Genentech, Harvard and Columbia University. There's currently also something going on with Stanford. We also had a project in Australia, and some projects are also, I think there was also something in Japan before. Now, the company covers quite a lot, like I said, from like model development, software engineering, custom segmentation, from like liver tissue over brain tissue, Lewy bodies, for example, or A-beta plugs, and also spatial biology. But one point I want to talk more about today is annotation service. So how does this work if I say 15.9 years of annotation time? Well, the company is 10 years old, and these numbers that I gave you are for models that are not older than 3 years old. So it's basically an annotator team that is available. So this work was distributed on around 30 annotators, and then you get down to like 6 months annotation time. And this is for our, I would say, 4 most important models for segmentation. We have currently an annotator team of around 70 people, and they are all biologists, and they work from ground truth in expansion microscopy, electron microscopy, light microscopy in different tissues, everything from liver to brain. And some of them work on ground truth, some of them work on proofreading. And so just for like expansion microscopy, but also electron microscopy data, we delivered around 25,000 proofread neurons in the last 2 years. As you see here, we can do, we can segment and annotate on various kinds of data. So we worked with this typical pan cell staining that Ons Emsad from Jörg Beversdorf developed. We work with other Lycon data. We do skeletonization, for example, on Lycon data, but also painting. So there are different varieties where we can support you with your science. It also doesn't matter like where do you can host the data. Also, we have our own solutions. You can bring your application. There are different varieties how you can work. Just that you have a number, our internal hardware scales towards the petabyte range. So 5 dimensions, multiple terabytes per data set is definitely not an issue, but is rather our daily business. Now, if you decide to work with us, how does this start? We basically define your requirements. You will talk with our management team, who are all biologists, so we understand your data. We define what output you want, like quality and throughput. Of course, you say you want the highest quality and the highest throughput. I know that, but the question is there always to find out, okay, what do you reliably need, or how fast can we do your throughput so that you get the quality needed for the analysis you do. This is something, to put that in more practical terms, we have customers who say, I want like 4 hours proofreading per neuron. That's enough for my analysis. I want rather throughput, so you cut it at 4 hours. And then we train our annotators, scale up your project, and then we can produce. To give you an idea, like about size, we have projects that run with 2 annotators, we have projects that run with 30 annotators. And for a ramp-up time, we can start within 1 month for like small, or we can start within 2 to 3 months for big projects. And what I mentioned before is, in our case, data size does not matter. You can bring us terabytes. Our company comes from the electron microscopy side, so since the beginning we work with tiled data sets. Our softwares load the resolution that's necessary, and so we can really work with any dimension. This is just to show you that we work with customers from all around the world, from universities like Oregon Health and Science University, but also from companies like Canopy Biosciences, or today it's Bruker Spatial Biology, and also with University of Tübingen, for example. If you have any questions, my colleague and I, we are available at the booth. This is only a short excerpt of what I showed you. I heard many people today talking about neurodegenerative diseases. I would love to talk about these topics with you, because there we have quite a number of solutions. That's our focus at the moment. Thank you very much.
Thank you so much for being on time. Are there questions? In any case, you can find them at the booth. So I would say that in the sake of time, we proceed. And we thank all the speakers for this session once more.
So, yeah, I'm the chair of the last session today. My big pleasure to announce Ilaria Testa from Stockholm. She studied in Genoa and did her PhD with Alberto Di Aspro, applying single molecule biophysics to fluorescent proteins. And then she moved as a postdoc to the lab of Stefan Helge here in Göttingen. And since 10 years, she's now in Stockholm and is a full professor since last year.
Good evening. Perfect. Thank you, Jörg, for the kind introduction, and thank you very much to Silvio and Ali for having me here today. It's really a great pleasure to be back in Göttingen. I'm really impressed about all the development in expansion microscopy, and myself. I still think that it has been really great what it can bring to 3D imaging, and we started the project back then to combine expansion and STED because I couldn't resist to see at this high level of resolution, especially for investigation in tissues and so on. At that time we were characterizing the lamprey and see there different cell types and being able to see the cilia symmetry, for example, in these cell types, which is still very difficult with common 3D super resolution methods. But you can do now that much better than we did back then. In fact, we changed focus as a lab, and we are very much focusing on studying another parameter that perhaps is still quite difficult to do with expansion microscopy. That is the study of dynamics, especially at small scale, both spatial and temporally. So I would like to focus your attention on a type of dynamic that, in my opinion, has been a little bit underlooked, which is rotational diffusivity. Typically, when we think about dynamic, we think about translation, single molecule tracking, or ensemble dynamics, time-lapse imaging. But the rotation can provide a lot of interesting information about molecules, how they bind, how they dissociate, and properties about the solution. So, but how do we measure rotation? Typically, if you have a fluorescent molecule, you can illuminate that with polarized light, and you have a preferential direction in which the molecule will absorb, that is the one parallel to the transition dipole moment. Then the molecule will relax, and depending on the relaxation, you can detect that with, for example, polarized beam splitter, you can say something about the state of the molecule. So you can photoselect that with polarized light, and then look at the relaxation or the scrambling of the dipole over time. Now, if something happened to the molecule, if it binds to something, then the dipole will scramble slowly. If the molecule dissociates, for example, it will be faster. And so you can measure that, for example, with fluorescent protein or different type of molecule that you can express directly in the cell. So in this sense, you can, in real time, record this type of data that are dynamic data and see and study, investigate the different state of molecule. So you will see a lot of this curve today. And ideally, and this is a dream, you can think about, measuring and track this heterogeneous population in cell over time and see, oh, this drug worked really well and it can increase the association of molecule while this one push dissociation. Right. So what I've been telling you now is nothing new. It's what is called a polarization microscopy of time-resolved fluorescence anisotropy. And it has been described a long time ago by Weber, for example, Lacowitz. And there is this nice relation that is the Einstein-Stokes relation that tells you how rotational diffusivity is so sensitive to change inside, so to the hydrodynamic radius of the molecule and changes. So at the power of 3, just to give you an idea, if you look at translational diffusivity, that is what we do, for example, in tracking, is only R. So you're very sensitive in change in mass. Right, so if it's so great, why are we not all using that to study molecular formation, complex formation in cell, or, for example, condensation? So the reason is that conventional time-resolved anisotropy is done with fluorescent molecules that typically have a lifetime between 1 to 5 nanoseconds. Now, this time windows is really short for a big complex to move, to rotate. So we will probably look static in your system. So then here comes our idea, that is, or our aim, that is to push what time-resolved anisotropy can to basically a bigger population of mass. Because if we see here, so this is basically what it can do with conventional fluorescence, and here is basically the size of the human proteome and its complexes. So we're quite shifted. So basically, if you want to study the association of a complex that is bigger than a GFP, you cannot do with normal fluorescent time-resolved fluorescence anisotropy. So and then here comes then the idea that is like to measure slow rotational diffusivity. And how we addressed that was with the use of photoswitchable protein. Now, I've been studying this protein for a very long time in the field of super resolution microscopy, but here we use it in a different way. So what are these proteins? Like, for example, you have the reversible switchable green fluorescent protein, you have DROMPA. Basically, you have proteins that are typically in one state, and then with light, you can switch to another state. For example, inducing a cis-trans isomerization of the chromophore, protonation, deprotonation. And then you can do that reversibly. So how comes that these tools are important for rotational diffusivity? Well, you can think of shifting the problem of kinetics to lifetime, that I said is very fast, to photoswitching, that are type of kinetics that you can actually change depending on the light dose. So you can change the rate of on-off switching, but for example, changing the intensity that you shine in your sample. So one example, but this is just one type of implementation, is like shining some 4 or 5 nanometer light to your sample, induce a conformational change of only a selected group of proteins, and then follow that over time. So then now you have all the time of the world to actually probe this molecule and get all the photon out. So you can increase the temporal windows in which you can actually track rotation. And so here is some example of how the data would look like. For example, if you would track the rotational diffusivity of a molecule that has a hydrodynamic radius of 50 nanometers. And that how it would be, for example, have 25 nanometers. So they're sort of well separated. Now, these are actually real data. We recorded that on beads of different size, of known size, and we anchored the protein, the GFP, on the surface. So now if you fit this curve with simple non-exponential, because this is a homogeneous solution, you can then actually find out the size, and this is what we knew about the beads, and that's what you can quantify. So they're quite in agreement with the size of the distribution. So it can be quite quantitative. Now, of course, you might not be interested in measuring only bit size, but you want to study something in a biological relevant sample. So in this case, we team up with virologists in Heidelberg, and they made this HIV-like particle expressing the reversible switchable protein in the GAG lattice. So now the protein is anchored to the lattice. Now, if the virus is mature, the GFP is cleaved away, so it rotates in this crowded environment. If the virus is immature, the GFP is still in this anchored in the GAG. So since this protein are also compatible with super resolution microscopy, we also took some resolved images of those, so we could characterize the size, but only by the pure quantification of the size. We couldn't really pinpoint for every single particle what was mature and immature. So we couldn't access the maturation state. But then by taking the very same photons and then analyzing that with a polarized beam splitter and analyzing them over time with specific pulse scheme, we could see that actually you have the faster rotating one here that are the GFP in the mature virus, and the one anchored in blue in the immature that rotates slower. And you can do that also on the single particle level. So you get enough photons to differentiate then from one population or the other. Now, you can also do that in living cells. Of course, in all this technique, you need to minimize the local motion of the probes. So, for example, you can express your RSFP with a smaller linker that is fused directly, in this case, with the alpha helix, the terminal alpha helix also of the histone. So in this case, you have a bit more rigid GFP. And then you can express that. You can see the signal here, the intensity of the histone label fused with the RSFP in the nucleus. And here you can see the different rotational diffusivity map. So now, to see if this is really like chromatin being more or less compact, we did this simple experiment where we had the control, and then we deplete ATP. So we saw that actually the anisotropy went up, and then we removed the medium, we recover, and then it went down again. So you can do this type of study and see and track, for example, what chromatin fiber more on the size beyond, let's say, the single nucleosome. So here we are talking about maybe 80, 100 nanometers, how flexible they are. And then you can do also this in different cell type, in different cellular state. Right, so another application where I think this method can be useful is the study of condensate. So here is Martina that fell in love for a protein named ARC, which is an activity regulator in neurons. So when we express the full-length ARC in cells, we could see the formation of this cluster of different size. So then there was a big question, how is actually the ARC organized in these big clusters? And that's where we did the stars imaging scheme. And then we could see that actually we had a signal, and this signal was speaking quite clearly to cluster size of around 80 nanometers. And that was really good for us because it also makes us think about the strengths of this method that is not actually sensitive to concentration. It can actually work even in very crowded environment. Unlike, for example, FCS that rely on fluctuation, so you have to be very careful to work with the nanomolar concentration. Now, if we then create a mutant, so we remove some part of the molecule, we see that there are still some small cluster, but this big one like disappear, and then also follow up with like no anisotropy signal, so we don't see anymore this low diffusivity. And this really gave us the courage back then to then go to neuron and then apply super resolution microscopy of the different mutant and then see that some can cluster, some not, and also in slice, and some actually of this cluster colocalize with RNA. But very interestingly, so we saw that when you actually overexpress the mutant that impede, so doesn't have impede ARC to actually form this large-scale oligomer. So you see here that is relatively homogeneous. You also have much more AMPA receptors. So it's really important. So this large number of scale oligomers are really important to actually control the surface level of AMPA receptor. Now, coming back to the method, as I said, so most of the application that I've shown has been done like in this way, where we photoselect the protein in one state and then we let it relax in another state and then we follow it, which we think is still very much probably the most quantitative method, and we could apply the very same theory of conventional time-resolved anisotropy, and it works quite well. But of course, you are changing state, so there is also some relaxation coming from a dipole rearrangement. So we thought it could be actually interesting to explore also other way to gain this dynamic information. So then we came up with this method where we basically switch off the molecule and with polarized light, and then we record these curves that are still analyzed then with a polarized beam splitter. So then the shape of the curve will change and will look like this, considering that basically the protein that has the transition dipole moment parallel will switch off faster than the other one. So you still have some information about the relaxation entangled with this method. And then, of course, we had to develop a new model to do that, and basically the rotational diffusivity will still be encoupled with the kinetics of the protein, but it works. It's still quite quantitative, and the nice thing is that here you get a lot of signal, so it's very sensitive, and this was the method that let us, that enabled us to have information on the single viral level. You can also have this very simple method, and thus we did it because everyone can do that in the lab as long as you have a pulse laser for 4 or 5 nanometers, but you can have a pulse diode, for example, and do that. And then you just change the pulse between these two 4 or 5 nanometers, these two on switching moments. So you change the delta T, and then you record how the intensity vary. And this is also connected to the rotation, right? Because if everything is static, then you would have a certain increase of intensity, but if everything rotates, of course, you don't have increase and so on. And then you can record this type of curves that are also representative of different sizes. So we really think that, we are really at the beginning of the concept. There is still a lot of work to do. And the two directions that excite me are, one, the big question, like how quantitative can we be? Can we actually, do NMR with fluorescence? Can we get there? But of course, we are at the beginning, so we had to do things. And the other one is more like, how fast can we be? We can actually use the real strength of this method, that is the fact that it can be scaled up to, answer yes/no questions. So what's the best way? What's the best component or cellular condition to achieve, more or less aggregation? And so in this technology that Yu and Guillaume has been working on, basically go more in this direction. So what they have been doing was moving stars from, let's say, the academics, is the 1.0 system, to this benchtop instrument that is very compact and compatible with multi-well plates, and based on a parallelized also system. And there we could, this optical development move our acquisition times from 5 minutes per cell, because before we were still scanning and getting information for every pixel, to less than a second per cell. So now you can think that you can basically then record many cells at the same time. Here you go, and you record this type of data of many cells, and then for each of them you can get your anisotropy signal. So this is like, again, the chromatin population, and then you can do the ATP depletion here and also see change, but more on a larger number of cells. Of course, to get there has been a number of technical things that we had to solve, including getting temporal information in the wide field system. And if you're curious, I can talk more about that. But so the direction here is really like to get then condition per cells and then try to create a map, an isotropy map that looks more sort of like fax data, and then from that, so we basically aim to reach almost this throughput, right? And that's sort of the vision for this method. Right, so I like to change gear now and talk about another type of dynamics that is more conventional time-lapse imaging, but done with super resolution microscopy, which we know that is not easy because we need more intensity. You have an increased photo bleaching, increase also like you have slower your recording time, slow down, and so on. And sometimes it can also be phototoxic because you need additional light to induce your switching or depletion, right? But what I really think is that automation can really help us to kind of start a new era in time-lapse imaging with super resolution in many ways. And for automation, I don't mean what I just talked about, that is basically asking the instrument to do repetitively a task that a human can do, just all over again, overnight perhaps, and this is how I learn about automation, but really use real-time image analysis and computation to move the microscope from a very static instrument to something that is sample adaptive and change, for example, resolution or speed depending on the need of your sample. And this idea started back then because, of course, I was in Göttingen, the hub of molecular neuroscience, and I've seen a lot of like those movies, right? Try to record the trace of synaptic vesicle, and I really saw how STED was actually capable of doing that very well. But still, like the throughput was not that high because it's difficult. I mean, we know in neuroscience that only a subset of synapse, fire, and then this firing, happen in a few milliseconds, and then maybe something that started changing protein recruitment and so on, then very rapidly go to base level. So you have to be really fast and try to detect when things like capture this time lapse when it matters, like in this transient moment. And capturing this transient dynamics is really not easy, right? Because if you would do that with STED, you would search for your calcium spike. It would take a very long time because it's a point scanning technique. So the recording time scale up with the field of view, right? Quadratically or even cubically if it's a volumes. And this happened really fast. So then we thought, like, how can we solve this multi-scale problem? And one idea is like, to push this event trigger microscopy that is basically using two different microscopes in one and use wide field microscopy, which itself is the most simple microscope you can think about, but it's still really, really fast, right? Very efficient. And so use that to scout for example, rapidly abrupt increase of intensity, like what happened in calcium imaging or even now voltage imaging. And then when that happened, only in that coordinate or around this start the STED time-lapse imaging on a protein that you care about. But still in this very tiny temporal windows. So here is Jonathan that basically had the wide field system on a STED. Of course, there has been some trick since there are two very different image formation to be really sure that the coordinate we were pinpoint in wide field really match with what we were scanning instead and so on. And typically we do the wide field in the blue. So we excite with the blue, so green light, GFP and so on sensors. And then we do the STED in the red because it worked very well and is also less phototoxic. like to have this rapid exchange. We have been working on a lot on the software. So Xavier developed this ImSwitch a long time ago that is a very, very interesting software to control scanning system. It's built on layers, so it really can control different complex devices without crashing. So many labs already adopted that, using that for MinFlux, Lightsheet, and so on. So really, if you're starting your development, you're really looking for an easy software, an open source software, and so on. I would really recommend, because there is a lot in GitHub. Also a lot of different versions and so on. And also he worked on the graphic interface to make that approachable also by users. And together with Jonathan, they create this widget that are standalone widget that you can basically, where you can modify a number of parameters depending on the event that you want to track. Increase of intensity, slow increase, fast increase, and so on. So typically with the wide field, we then record many, many frames, and then at a pair, we do this ratiometric imaging with pinpoint where the calcium spike was, and then there we start studying like a synaptic protein, like synaptotagmin, using also all the fantastic tools that Silvio, Eugenio, Felipe has been developing over the years. So we could see that if we just quantify the speed in this synapse, just a few milliseconds after the calcium spike, we measure, for example, a different displacement than if you would randomly pick up the other, which point us to the fact that a lot of things can be then masked by averaging. So if you are not timely in recording them. And then, of course, you can push the temporal resolution if you want. You can decrease the region that you are studying and then go basically real time. This is another example, and this is one of my favorite examples, where we are triggering the STED on another sensor, a pH sensor, that in this case is expressing with the CD63, so exocytosis. So this is a tetraspanning. So where you see that, you can detect, of course, these events like this one that are there, take a long time, and this is probably what you would detect more likely if you wouldn't have the event triggering, right? But then you have also on the right side these very transient events that are popping up. And then with the STED, if you start labeling the membrane, so the lipids in this case, and you see them side by side, so now you're looking at the membrane on the side, you see this nice bending, and then you can see the little one, like, the very fast one, whoop, happening there. So this is exocytosis. And here you see better, like the bending of the membrane, the different moment for the two events, the fast, let's say, exocytosis and the slow one. This is another type of triggering where we use, again, the wide field to track this basically exosomal protein, this tetraspanning in the process of the neurons. Of course, you don't need super resolution if they are sparse, but then when they start getting together in the maybe checkpoint, that you're wondering, well, what is there? Is there a fusion or not? And then you can switch on the STED and then again resolve the lipids and then see how the structure look like. For example, if they are like spherical or not. In this case, they look tubal-like, and it doesn't seem that there is a fusion in this case. Then they go separated as a two different objects. But this is to me is really exciting. We had this just three different type of triggering, but this is just the beginning. There could be so many other ways, right? So many sensors around, so many cellular processes to look at. So this is really to make your recording more efficient and perhaps faster. Now, there are also obviously time-lapse or biological investigation, dynamic investigation, where you cannot deal to work with very tiny field of view, but you still want to have your context and perhaps you want to resolve that. And so this is why we have been working for a very long time on a technique named Resolved, which is a sister technique of STED, as a concept, wanted to generalize what STED was presented to, and the idea is like the image formation basically is really similar to STED, the idea of using light patterning, but instead of stimulated emission, you use reversible switchable fluorescent protein. So basically, instead you deplete all the protein around the focal spot, typically with a donut, inducing this transition, while with Resolved, as we've seen before, you can switch off the molecule, so change conformation so that the one in the periphery of the focal spot do not emit. So why it's different? You can do that with less intensities, and of course then you need though to use this specific protein, so you cannot use any proteins, any probes, so they have to reversible switch, and they have to have specific kinetics. They need to be able to switch on and off, and when they are switched off, they have to have a little background, right? So there are like green and red protein that has been developed and can work. So, but now the beauty is, since you just need very little intensity to switch off the periphery of the focal spot, you can do it in multiple points. And that's what we call it parallelization. And one of the first paper was, we can do that now with 1,000 donuts. Well, so resolved. And now the point here is that instead of moving one being across the field of view, which would, of course, where the frame time with scale with the number of pixels, you just illuminate multiple points at the same time, so you just scan a little region, and then you get the rest for free. So it's really faster in large field of view. Now, we call it multiple donuts, but in reality, we never use multiple donuts, or at least not yet. What we have been using are standing waves, very similar to what has been used in structural illumination, but compared to what has been used in linear structural illumination, we need a very high modulation. We need the zero to be really zero. Now, for 3D, we developed this relatively new light pattern where we are also depleting above and below the focal spot. And compared to 3D sim, now we want every zero to be the same and to be equally spaced, because we want the image formation to still be very simple. And that's what we use to switch off the molecule. Now, you might think that if you have a cell that is full of a RSHFP2, now you're shining blue light with this type of honeycomb, so all the protein then above and below the focal spot will switch off, and then you will create these nanovolumes. But now you're not just creating one at a time and reading them out, but you are like imprinting that all over the volumes, and then you are reading them out. So then you can get basically isotropic resolution. So we used that. Initially, we were still reading out plane by plane, but then we have in the latest configuration, we combine the photo switching with a very fast sheet of light, oblique plane illumination. And then in this case, basically, we parallelize the switching. So we switch the protein in one step, and then we read them all out. And this is also very important because you minimize the demands on the protein. Now you don't need the protein to cycle that much, so you get the best out of it because you are not wasting cycle that happen above and below the focal spot. So with all this optical development, we move from the very first implementation of Resolve that was based on the donut, where we still could get nice resolution, but was very much confined, to this implementation that is based on the standing waves and then multi-spot to read them out. And we call it Mona Lisa, that we have in the lab, where you can get quite large field of view at the very same frame rate. And then if you use the latest implementation, that is the one combined with the light sheet, you can get volumetric reimaging with the very same frame rate. And then you can look at cell division, or you can look at mitochondria motion. I think my computer is getting slow. And here, like some examples. So if we want to have a close look, then on this data, you can basically see all the very nice features that would have been, hidden in the very same recording, take it without the off switching. So you can really see the RSHFP2 localized in the outer membrane of the mitochondrion. And this is live, so you can record several of this volume over time. So you can, for example, apply perturbation. You can block the ability of mitochondria to do respiration and then see what happened. And it's nice because what we find out is that if you look at a very fast change, you have an increase of fission rate, but then you start then having all this collapse of the membrane, forming this sort of horseshoe type of structure, and then you start having also the budding out of this very tiny vesicle, which are reported in the literature to be mitochondria-derived vesicle and full of oxidated protein. Now, so this is great. So the ability to do like volumetric imaging, record different volume over time. But of course, there is always the question, you know, how long can you track your cell? So for, you know, how long can you track the signal before it fades? And you can tackle the problem is making the protein, let's say, more photostable, and that's what we did for many years. Or you can tackle the problem in making like better optical schemes where you waste basically this, try to waste less, to minimize the waste in protein photocycle. But then I feel that, you know, you start reaching sort of a plateau there, what is possible. So at this point, then we start to think about other direction, and then perhaps, you know, think about what deep learning can do and develop deep learning tools to improve our data. So in the way we thought that they could really improve resolve volumetric imaging data, is in two ways. So first, by enabling us to lower even more the on-switching intensity, and then to photobleach less, and then to restore the image. And the other way is like to tackle the speed, to get even faster, sort of beating the sampling theorem that we all have to follow, that is like the Nyquist-Shannon theorem. So the very first way is like, instead of doing, let's say, a resolved with on-switching intensity that typically it's selected to switch on 80–90% of the protein, because we want to see most of the protein typically, and then we switch them off and read out, to only use a portion of it, like 10–20%. And then of course the image will look noisy, right? But then you can restore that. And then use deep learning to do that. And this is very robust because you can build very strong ground truth things. You know, you always can take frames with the full on-switching intensities. So we have been teaming up with Florian and Arneban using their U network. They work really well. We test different networks. A typical problem in using this deep learning network for super resolution, they typically smooth the structure, so you lose resolution. But I think this HDN was really, really good in preserving the resolution, and that was important to us. And then we could apply them to our data, and you know, of course, the improvement was really, really great. So we could follow the same structure for like, you know, with many more frames for much longer time. Also because something to take in account, the photo bleaching of the 4 or 5 is not linear. So basically by using this low intensity, we impede the ResGFP to get stuck in this intermediate basically state before the relaxed state in the as their own form that bleach a lot. So then in this case we could really work in a range of intensity that is really gentle on the protein. So and these are some examples. So here you see actin label, here vimenting. And what I like this is that we could see, for example, the actin meshwork for a very long time. This has been an incredibly difficult structure to follow for more than 5 frames. So now with this restoration, we could follow for much longer. And here you see the same on vimentin. This is the cell line that Stefan Jacobs did many years ago. So it's a CRISPR-Nukin cell line. So you're looking at endogenous level of expression here of vimentin. Now, the second approach where we used the UNet for was to beat Nyquist. What I mean for that? So still, the single parallelized resolved frames is built upon like different scanning different images, because you still need to scan at least one small region, the diffraction limited region, right? So, and this typically you decide the step size based on the resolution that you want to get. So if it's like 50 nanometer, maybe you sample every 20 nanometers. So, and this is still obviously there, and it's what overall limit your overall frame rate. So, but what if we could, for example, only sample and only get a very small subset of these frames and then guess what it could be the final image. So, and that's what we use to. So we basically have, you know, like we train the network with data where we do, of course, the complete resolved system. And then in this, we have, let's say, 100 images of that. We just take, you know, 50% of this or 25% of this, and then we guess, we let the network guess what is going to be the final structure. And then you can do that in many ways. You can always take the same pixel, but much better is actually to randomize this process so that the network doesn't learn about specific pattern. And what we saw that was very useful for this approach and what really made a difference for us was inputting not single frames, but basically a set of 5 frames to let the network learn also the temporal evolution of the system. So this worked, was really the crucial point that made things working reliably. So then we applied that to our favorite system that are mitochondria, and then, so here you see, for example, again, a conventional time-lapse imaging taken with resolved, and then you see that in some cases you get, of course, motion artifact, and some mitochondria are also like this one, suffering a little bit. For the light intensity, and this is like the data restored with this pixel hopping method. So we could first of all see these tiny events that are really fast. You get this tiny protrusion of 100 to 100 nanometers that happen really fast, and you have to, you know, being able to look at the sub-second frame rate to actually spot them. And then you can see that for of course much longer, because you're also shining less intensity. So you have the two advantage combined. So here, for example, the number of events that we would spot in conventional resolved imaging, and this is like the increased number that we see now on the with lowering the intensity of skipping pixel. So you can see many more of these budding out events. And especially for our study on the right vesicle, this was really important because it enabled us to not only see budding in and budding out, but also understand the directionality of the vesicle. So where do they go, and if they, for example, fuse it again. And this is like one last example, because of course we can apply that to 3D imaging, so we don't lose the vesicle, and then we could see that, you know, budding out and then in again of the same mitochondria all over again, and increase, you know, the throughput of this type of data. Right. So that brings me to the end of the talk. So I've presented you like 3 different methods. You see 3 different types of dynamics and dynamic events, transient events, rotational diffusivity events, and more like volumetric sort of traditional time-lapse events, but at a smaller resolution. For that, I want to thank the group that has been behind this research, and of course the funding bodies for supporting the research. And thank you very much for the attention. Not worry. Yeah, so for the deep learning, right? So okay, let's start with ARC. So basically the big question, I mean, I haven't discussed all the biological part behind the ARC, but that was like the big question of Martina thesis, right? Like, are these oligomers, this capsid-like structure that has been reported in literature actually present in neuron? Because what people have been seeing that ARC is actually, if you purified it from bacteria, in specific conditions, it formed this beautiful capsid-like structure that can contain RNA. And has been reported that this is, for example, a method to, you know, pass RNA between, you know, synapse to synapse. And this what excited us back then to see, can we see actually this method with our tools? And again, at that time, there hasn't been, or even now, still tomographic data showing this capsid in neurons. And my point, our reflection at the time was like, perhaps it's a rare event, right? Because indeed, it's an activity regulator that happens, is transient, and that's why we don't see. It's not easy to find those. So then with Martina we did two things. First, with the endogenous arc, we tried to see it, and then by combining, so with STED we could always see specific clusters that have been pointing to a distribution that make sense to this number of molecule and size. But then we team up with Ralph, we did paint on those, and then you can see that actually are very ordered. So we think that those are the cluster. Still, they are not as much as we thought, so perhaps, I mean, the question is like how relevant is to have them in this specific order. But what we definitely saw that these large oligomers, independently if they are ordered or not, are really, really important for maintaining the surface level of AMPA receptor. So then, we move more on that side. But yes, but definitely. So ARC. I mean, we tested in many ways. Also with other drugs, it seems that to form phase transition, and this is also found in the synapse. So, and then the question is like how this is connected with this capsid-like structure. So the second question is artifact. So for artifact, like the first thing is like about image restoration. So in that, I think you cannot do much wrong, right? The worst things that happen is that you lose resolution. But this with an FRC or measuring, in a statistically relevant way the size of the filaments, you would find it out. So that I wouldn't say it can be a problem. For the skipping pixel, of course, there is a certain number, at least in our hand, that vary from structure to structure that you can skip, which is not, 90%. So, but we still that, for example, maintaining 25% of the information is still fine enough. Now, the question that is like, how good is your ground truth? And in our case, we made sure that we could record and see things that actually could be validated with real data. Just in this case, we get a much higher throughput, right? And for example, we had directionality motion. But the budding in and budding out, I mean, these are seen already in, sort of raw data. Yeah.
Hi, a very nice talk, and I'm interested about the first part of the talk about the rotational diffusivity. Do you have methods to manipulate the actual rotation of the protein? So I was thinking, have you tried to attach magnetic nanoparticles to the proteins and turn on a magnetic field to influence the torque of the proteins and see if it affects the dynamics of the condensation and so on?
Yeah, so we haven't tried that. I think is a very nice suggestion, but there are ways that you can somehow perturb the diffusivity, right? Just making your solution more or less viscous, for example, and this kind of thing. So we have been trying a few of those to see if it responds by slowing down and so on. So, but I think this is really interesting, and it's also really interesting to think about different type of diffusivity, not only sort of spherical object, but also like other type of things. And I think that, for example, like origami structure could be a very nice template to study that, because you can control the different shape and then develop different diffusivity model for this. Sort of learning of what NMR people are actually doing all the time, right? And try to apply to a very different system.
Thank you.
So there's another question here in front.
Yeah, actually also about rotational measurements. I have a great idea, actually, and I've gotten a lot of flashbacks to my NMR lectures. Now that we can probe this different degree of freedom, so we can probe the translational motion through FCS, and then we can get the rotational motion from here. In the condensate, have you actually seen that they track with one another, or is there maybe more rotational freedom than there is translational freedom in such a dense mesh of protein?
Yeah, it's hard to measure translational diffusivity for us there, because the intensity were really, really high. So initially how we started, we wanted to do FCS there, but then of course we could do that in the cytosol. But then what happened, we did then FCS in the cytosol, and then we did starts both in the cytosol and the condensate, and then we could see that they had a very similar liquid-like behavior. So then we can then bridge the information to the translational part. Yeah.
Okay, thank you.
Yeah, okay, are there other questions? Yeah, Mark.
Thanks for the very nice talk. Stars, it got me thinking, how many molecules do you need? How many fluorophores do you need to make a stars? Can it be a single molecule measurement? Or how many fluorophores do you need to measure?
Yeah, no, that's also a very relevant question. So we didn't push much to go to the single molecule level, right? Because then you would do classic, study of single molecule and then rotational diffusivity to see one by one. But I think the strength of this method is actually to see the ensemble, right? Now, still, you might have objects that are small, as the virus, where we label, for example, about 100 proteins, and that was fine. Can we push it to 30 proteins and so on? So that's something we are working on. Probably in a very simplified system you could, but we are not pushing that so much because I think that is rather the contrary, right? To try to see you want to be sensitive, of course, but then at the end you want to see the whole population, right?
I'm thinking about heterogeneous samples, right? Is there some mechanism that you're thinking of to see mixtures of different populations?
Yes. So we are working on that, and then of course we started with the more control system like the beads, and then mixing beads of different types and see, at the end when it's 1, 2, 3, 4 is a linear exponential that are summing up, right? So this the problem then is not that big, but when we become multi exponential, that's another story. But typical things like I have protein in solution and then I have 3 different types of aggregates, and we mimic the beads to be that, and we are playing with the concentration. That is still very much feasible with very simple tools. So I believe that even then, more with AI or this type of method, then you can also try to approach more complex situation, and then dream about the mass spectrum of a cell, sort of.
Okay, I have further questions. If not, then I would like to thank Julien again.
So dinner commenced now. You feel free to go, but Chiara et al. We're meeting here in the room.
That's right. Thanks a lot. And then we will see everybody again tomorrow, starting from 9 o'clock. And the last chance tomorrow morning for the various social media events, so to say, and pictures and everything, and prizes in the evening. See you then. Have a good evening.